of tuberculosis. As we all know, today's session is based on the application of highly sensitive techniques used for biological research. Among these, today will be focused on flow cytometry and bioplex. Now, I would like to introduce our first speaker of the day, Dr. Ramsha Kumar, Application Scientist, Becton Dickinson Biosciences, New Delhi, India, who is going to give a talk on BD Rhodopsin Single Cell Analysis System. Dr. Ramsha is PhD from AIIMS and currently she is an application scientist for single cell multi-omics at Becton Dickinson Biosciences. She has more than 13 years of research experience in the field of immunology with expertise in HIV pathogenesis and chemokine receptor signaling. She has an excellent experience in single cell multi-omics, cell sorting and multicolor flow cytometry. Now without further ado, let's welcome Dr. Ramsha to start today's seminar. Ma'am, please.
Thank you. I'm audible? I think so. Yeah. So it's nice to see some student in reality, because from last 1 or 2 years I'm seeing everybody behind the camera and I'm talking and I can't see anybody, how they are responding and what is their reaction. So it's very, for me it is very good experience and very nice. It's the first experience I'm having. From last 1 year onwards I'm giving all those lectures and everything in online mode. Yeah, as a speaker, but as a participant, yes, already I came several times when I was in it. So before going, because it's mostly before going in detail about this BT Rhapsody, which is a single cell analysis system for high dimensional biology research. So I just want to ask, how many of you have used flow cytometer? No, it's okay. So the thing is, I am saying that the title of my talk is BT Rhapsody Single Cell Analysis System, a robust microwave-based single cell partitioning system for high dimensional biology research. So the question first, I'll talk about BT Rhapsody. So it's a new machine which BT has launched. For single cell analysis system. So that I will show. But the last line of my talk is for high dimensional biology research. So what do you think about as a high dimensional biology research? Any one of you can, I want it should be a little bit interactive. So if any of your student can tell me, do you understand by high dimensional biology research? Yeah, yes, like that. So in flow cytometer, yes. So when you do the flow cytometer, you look all the surface markers. So this is one dimension that we are looking the protein markers, how my cell looks like, whether it is a CD4, CD8, macrophage, what kind of cell. So this is one dimension of that cell. If you were talking about the intracellular cytokines, now again another dimension on the basis of which I am going to, which will give me what would be the functional aspect of that cell. So in that way we are giving it to the second dimension. Now I am saying high dimensional. So what is high dimension? Now beside this protein, there are the most of the things that is you people, if you are working on molecular biology, you know that we have 20-30,000 genes in human and same like in other animals. So if we are going to integrate our data, that is the gene data, all the transcriptome data with our protein data, so that think about a picture in your mind that you are giving another dimension of your work, your research work, and then you are going to analyze it. So you are getting a whole picture, a comprehensive thing about that this is about my cell and how it looks like. So to identify any novel cellular targets, like any novel cell. So suppose you are working on any disease like COVID. When COVID came, nobody knows that what kind of cell, what kind of the immune system it was what is that kind of cell responsible for a person is asymptomatic or severe, or it is having no symptoms or a mild symptom. So what are these immune cells, how they are playing the role? So you will take, if you are a flow cytometrist, what you will do, you will have some marker based on the complete blood count, that my lymphocytes count are getting down, neutrophils are going up. So you will have some marker. That is your judgment, that I'll use this 10-15 markers and I'll check this is the data. But when I talk about gene, is it anybody knows before that what kind of gene, when a person is severely COVID infected, what kind of a gene he is having, why it is that? So these kind of a data you will get it when you will integrate both the data. I wanted to convey this message that when you integrate your protein and gene data together, you are getting something new, or which is unbiased, which will give you our cellular targets, or which will reveal, which is important to resolve any cellular heterogeneity. So the thing is that to resolve any cellular heterogeneity, and we used to use this protein and gene data together. Why? Because as per Nature paper in 2013, that mRNA and protein data are not always correlated. So it means that if lots of mRNA is present, it doesn't mean that that amount of protein is also present. So because of this post-transcriptional and translational modification during the time of development, and because of any pathological situation, this protein and mRNA data is not always correlated. So you can see a very beautiful quote is here, that in biology, nothing is clear, everything is too complicated, everything is a mess, and just when you think that you are understanding something, you peel off a layer and find a deeper complication beneath that. So it is very simple. So that's what I wanted to say, that you have to if you wanted to resolve any cellular trait, if you are working on any cancer cell, the cancer tumor environment is the best environment to understand about the cellular heterogeneity. One cell is different. See, here is a cancer, this part is cancerous, but the nearby cell is not cancerous. So what is the factor why this cell is not cancerous, and how it is different from that? Sometimes in some patient the tumor, if they are going for any therapy or that, in some patient it is very successful, in other there is a recurrence, the cancer therapy is not working. So everything behind that is there are some genes which play a vital role in identifying or in the therapy and everything. So what is our normal approach as a researcher? Most of you people, to understand the protein expression, we do microscopy, mass spec, flow cytometry, or either we do qPCR or RNA-seq. So now the BD has come with this new technology, new machine, which is BD Rhapsody. Okay. This is a Rhapsody machine. I can't see it. It's a scanner, and if you see this one, this is an express system. Okay. So I'll go in detail about this, what is this scanner do, and how this express help in enabling you to have a single cell data. So what is our offering is, like we have this machine, Rhapsody scanner and express, and these are now you will and we have these many kits. So kits or agents which help you to start it from a single cell to the library preparation. So when you prepare the library, it is ready to load it on any of the Illumina platform, NovaSeq, NextSeq, anything. So we have the reagents for all those things, and the question generally as a biologist, anybody can think about that, oh, I am going to get it the single cell data. How am I going to analyze it? Because I am not from bioinformatics background. So most of the people are not from bioinformatics background. So it's always an apprehension that I don't understand Python and R. So how helpful it will be? I'll have the data, but I'll not be analyzed. So there is again a solution for that. So we have this pipeline which is known as 7 bridges pipeline, where you have to upload the data, and this SIGIC software. So if you have used Flojo software, the same way it is like the SIGIC software, where you can analyze You can visualize your data, you can make Disney plot, you can make UMAP, volume plot and everything with the help of the CK software. So we have end-to-end solution of this technology from the single cell to the data analysis. Just a little bit about the what is this Rhapsody? So it is a scanner, and this small instrument is called Express. So we have two configuration. One is Rhapsody scanner with Express, another is only Express. So what is scanner does? It gives me the visual workflow. So each and every step it will tell me how many cells I have loaded, what is the viability of the cell. So it has some lasers that will tell you that what kind of, whether if you have loaded 10,000 cells, so whether how many cells are viable. So that also it will give you, as well as it will tell you what is the multiplet rate. So because I am talking about single cell, so I expect that my data should be single cell data, not there should be more doublet. So it will take care of that. It will tell me at each and every step how I'll show in detail that how the visual workflow looks like, and it is recommended for users who are new in single cell workflow and if you are working on any novel cell types. Now this is a small instrument which is known as Rhapsody Express, and it is for those users who already well versed with the single cell technology, and it's a very small instrument. You can keep it inside the hood or anywhere. Just have these two magnets are over there, and there is a place where you can keep the cartridge. So if somebody is working only on PBMC, then they recommend to use only Express. Otherwise you have to use scanner and Express both to get a visual workflow of the data. So how this system how it is works, so it's just an overview. First we load the cell, I'll go in detail. First we load the cell on the cartridge. This is the cartridge where we load the single cell. Then we load the beads. These beads are not normal beads, these are magnetic beads which are barcoded, and then we lyse the cells and all the mRNA will come out, because I'll tell you that our capture mechanism is through magnetic bead. So all the mRNA which has come out from the cell, it is going to bind on the bead. Then what we do, we collect the bead with the help of magnet, and then we synthesize the cDNA from the RNA, and with the help of the kit we made all the libraries. And then we can visualize the data. So we do not use any microfluidics. Cells will settle into the microvels by the gravity, and there is no fluidic pump is required. So just a minute. So I was talking about that we have this reagents. So what is this? This is if I am saying that you are going to analyze both protein and gene data. So we have this kit which is known as whole transcriptome amplification kit, where you are going to have the whole transcriptome data. So that will take care of your gene part, whole transcriptome amplification. Targeted kit, where you will have, there are different kind of a targeted kit. Like if you are working on human thing, then we have human immune response panel, we have onco panel, T cell panel. So there are different kind of panels that we are having. So we have this targeted kit also. Now I told you that here we are going to integrate both protein and gene data. So when you are doing flow cytometry, how do you look protein, like if your protein like CD4, it should be tagged with any of the fluorochrome like FIT, P or anything. So when you stain your cells, then you go into the flow cytometer, you can see that this is my population of CD4 or CD8, anything. But depending upon your flow cytometer, this thing that how many lasers it has, you have a limitation that you can use up to 10 color, 20 color, 30 color, or maximum in India up to 50 color. So this is, you are going to use it, the antibodies. But here we are going to use the same antibody, same clone of that CD4, CD8, but here it is not tagged. This is called AbSeq. It is not tagged with the fluorochrome. It is tagged with the oligos. So that is why it is called AbSeq. So antibody is same, but the tag like FIT, P, it is tagged with the sequence. So it is we use this AbSeq technology to identify this protein expression. So now I have covered that we can check the gene as well as protein. Now what else is there? It is sample multiplexing technology. Like you must have known that we can barcode our samples. So how do we barcode? So same way we have this kit which is known as sample multiplexing kit, where you can barcode at least 12 samples together. You can pool it and it will become one sample, and you can use this sample multiplexing kit. And we have another kit which is known as VDJ, which is for to identify the full length of immune repertoire analysis for TCR and PCR. Now, just I told you that this is a general structure of an antibody, you know. So, if this antibody is tagged with the FITC, so the FITC will be tagged here. But if I am saying that this is an AppSeq, so this is the oligo sequence that our antibody is having. So this is again having this poly-A tail. What else is there is antibody specific barcode, because how do we identify later on? Because in flow, when you do the flow cytometry, at the same moment when you have the plot, you identify this is my CD4 population. But here you will get it only after the sequencing. So how do we identify? Because of this antibody specific barcode that every antibody has it, and we have this unique molecular identifier on that, and the PCR handle for library preparation. And I told you that we are going to use this magnetic beads. So how is my magnetic bead? So this is the magnetic bead, which has the short DNA fragment immobilized on the bead to capture the RNA from each cell. So you can see this one DNA fragment. If I will say this is the elongated structure of that. So in the tail it is having oligo-TT. So our capture mechanism is through this oligo-TT. Every RNA, whatever it will come, it will have the poly-A tail, and this bead is having oligo-TT. Our AppSeq is also having poly-A tail. So then everything is, whenever the RNA is there and we are going to put the bead, so all the RNA will come and it will bind to this bead. Beside this, the bead is having cell labels, unique molecular identifier. So cell label will tell that which mRNA belong to which cell, and this UMI is to check the PCR biasness also. Now I told that we have the sample multiplexing kit where you can barcode your sample. So again, this sample multiplexing is also having this poly-A tail. And why it is important that there are 12 tabs, and this DNA barcode acts as a RNA to mimic by 3' RNA sequence because of the oligo-TT. Now what is the why it is beneficial? See, when you go for the sequencing, how the sequencing people charge, they said your one sample will cost this many. But now, if I am saying that I have pooled multiple samples, now it has become one sample. So your 12 sample will become one sample, and you have to pay only for one sample. So in that way, you can reduce the cost of sequencing, you can reduce the batch effect and other thing also, and the technical. So multiple time, like if you wanted to, suppose you are working on any cancer, so you wanted to have the same patient tissue, same patient blood, and some follow-up data also. So if there are 4 samples, like tissue sample, then PVMCs before therapy, after therapy, and you wanted to do it in one experiment at a time, then you can pool tissue, blood, everything by barcoding, and then it will become one sample, and then you can stain it with the AppSeq antibody, and then go for this cell lysis procedure and to isolate the RNA and then make this cDNA. So this is our cartridge where we load the cell. Here is an orifice, it's a U-shaped structure, and this is a honeycomb kind of a structure. That's why I said that VD Rhapsody is a microwell base in the cell partitioning system. So this is the microwell where each cell will go and settle, and this is the scanner, and this is the magnetic bead. So I'll show you, there are more than 2 lakh microwells in this cartridge. So how we load the cell? You can see these green dots are the cell. They will settle down through the gravity. Now the question is, how do we load the cell? So we have the automated pipettes which optimize the flow rate and which ensure that in each well only one cell go and settle. And sometimes it happens that there will be some wells, there will be 2 cells that can settle. But the bead is like that, in one well, only one bead can go. So that ensured that one cell, one bead. So that ensured that low multiplet rate. Now after that we load the bead, which is the magnetic bead having oligo-DT tail. Now the beads will go and settle. We load the bead in excess to ensure that each cell will bind to the bead so that the capture rate should be higher. Then we wash off the excess bead. This is the enlarged structure of the well, where this is the cell, this is the magnetic bead. There is no interaction. Then after that we lyse the cell by using lysis solution. All the mRNA will come out and it will bind to the magnetic bead. And you can see this in this way. This poly A and this RNA is going to bind to the bead, and then we collect through the help of magnet. It is inside this Rhapsody Express machine. Then we collect it into the append-off, and then in that append-off, the bead is having all the RNA. Then we made the cDNA, and with the help of, like if you are working on a whole transcriptome amplification kit, then we made all the libraries. So the kit is containing each and every reagent which is required. No, the beads cannot be reused, but bead can be kept for further. Like if I have captured 40,000 cells in that bead, I made the library, and I did the sequencing around 30,000 depth for whole transcriptome. And I came to know there are some genes which can be amplified. So can I go back to the bead? Yes, that bead can be saved, and then again you can, from that bead you can make the library and you can do the sequencing for more depth, or if you wanted to do some other kind of study. So beads is already always be having this, all the barcodes and everything is there. So I was talking about that our machine give the visual QC. How? Like this is the only machine in throughout the world which can give you visual workflow at each and every step of the single cell data. That it will tell that number of wells with viable centers. So if I have loaded 10,000 cells in my this thing, it can also count. Also there is a Newport chamber in that machine also, where you can count. And because we are loading some dice, which will tell me that my cells are viable and how it is. So if on that basis, it will tell me that how many cells are loaded, what is the multiplet rate, how many beads that captured to the cells, and again after that what is the multiplet rate. So at this step only, we can decide to go for the sequencing or not, because many times it happens after the sequencing data will come and we came to know, no, my data is like a crap data because most of the cells are doublets and most of the cells are dead. So when you see the dead cell, though there will be more mitochondrial RNA, so there is no point of analyzing that data. So at this step only you know that my cells are good and it has a good viability, whether to go ahead for the library preparation or not, and we also know that how many cells we have loaded. So when we are sending it for sequencing, what should I tell to the sequencing people, that this is one sample having 10,000 cells. So on that basis we can decide the depth of the sequencing, and we can decide for each and everything how much library I have to prepare and all. Like analysis, yeah, because yes. We have one more product which is known as Immune Discovery Panel, which is a panel of 30 antibodies. So if you don't know about, like this was mostly used at the time of COVID, when nobody used that, nobody knows that what kind of a cell playing role and all. So it has measure all those cells, CD3, CD4 and others with activation and absorption markers is there. So if you don't know about which panel I have to use, then you can use this 30 antibody panel. Otherwise, there is no limitation. There is a paper recently published that you can use up to 200 antibody in one sample. So there is no limitation. So in India, I have supported at ICGEB, where they have used up to 40 antibodies for this COVID patient, and recently we are going to use up to 55 antibody in one experiment, where the number of sample could be anything, but that vial come with for the 25 test. So see, imagine if you are using sample multiplexing, so you are pulling 6 samples in one tube. Now these 6 samples will use the antibody of one sample only. So you are saving the antibody, you are saving the cost of the sequencing also, and there are 6 samples, there will be no batch effect, no other thing, you can do it with the help of these technology. So this is again that I was explaining that we can use from 20 plex to the 100 plex. This is R&D data where you can, there is no, it is not compromising on the gene data. So if you are going to use 100 antibody, there this gene and protein data will be the same. So what is the advantage of having this microvel based single cell partitioning system of the BD Rhapsody? That it gives me a superior cell capture rate, low multiplet rate, highest throughput, and we can go beyond PPMC. As I told you that we can use tissue sample, we can use CAR T cells, we can use other kind of cells also that I will show and will give the reliable and consistent result. So what is the meaning of a superior cell capture rate? Means if I am loading 10,000 cells, how many cells are going to be captured by this cartridge? So that is more than 75 to 80% cells is captured, and that has been checked by using a mixture of Jarkat and Ramos in 1:1 mix, and then we have checked, this is R&D data, that it will showed that intra-sample variability is very less, as well as the capture rate is very high, more than 75%. And this is not the BD data, it is published in all other papers by using Rhapsody technology in all the Cell and Nature publication, where they have used different kind of cells like stem cells, myeloma cell, FACS sorted NK and T cell subset, or tumor xenograft or CAR T cells. Depending upon the tissue also you can use it. For mouse also you can use it. And what is the capture rate also it is mentioned here. So it depends upon a lab to lab what would be my capture rate and what would be the viability and how many number of cells you can load. So the question is how many cells I can load it up to in this cartridge. So as per BD recommendation, you can go from 100 to 40,000. But I myself for my experiment loaded 52,000 cells and the data was good. So I know that we can go up to 50,000. And there is a publication in Cell where they have loaded 60,000 cells on Rhapsody. But BD recommendation is up to 40,000 cells. So the thing is, like, yeah. See, the question is, if you are working on flow cytometry, so you will say, I am looking the protein, whether the same kind of thing I will get it from the using AppSeq or not. So this experiment was done. So and the bone marrow extracted from 2 mouse, and then it is analyzed on the BD FACS lyric and using the Rhapsody also. So you can see this orange one is diagram bar graph is for the flow cytometry data, and blue and gray was for the SCM, single cell multiomics using AppSeq technology. So it is comparable. All the immune cells, if you see the same amount, whichever it is showing by the flow cytometer, you will get it through the using AppSeq also. Now, just now I have told that we can pool multiple kind of cell. So whether we can pool different sizes of the cell or difficult to handle cell like neutrophils, you know that it is very sensitive to handle neutrophil. So in this experiment they use, you can see these cells are of different size, different properties, functional properties. Some is T cell, neutrophils, K562, every cell is different from each and every. So these are having different sizes, and the question is, if I am doing it one cartridge, whether I am getting the same kind of data if I repeat the experiment. So this is also done within 2 cartridges, different sizes of the cell, different properties of the cell pooled together, and then they have checked that how many cells have loaded, like 5,000 cells were loaded, and out of that how many cells were retrieved after sequencing. You can see that more than 80% cells were retrieved after sequencing for all this. So we can pool different kind of cells. That question we can handle easily. Now again capture rate. So capture rate, if I am using 10,000, whether my capture rate is 80%. When I go for it 40,000, whether my capture rate will decrease or it will compromise? No, it is not like that. So from 10,000 to 40,000, the capture rate is more than 80%, and the same is for the multiplet rate. But of course, at 10,000 the multiplet rate is less than 2%, but at 40,000 it is again less than 10%, which is very good for the single cell data. Now in this experiment, what they did, that T cell activation time course examined on the technical replicates. So this is again the T cell were stimulated at different time points, different days, and the sample tags that I told that we can use this sample tag for barcoding our sample. So see, fresh sample, 3 days, 7 days, all these samples were first barcoded with sample tag. Then they were pooled and then loaded onto the cartridge 1 and 2. And then they checked that the percentage of the cell assigned to each sample tag per cartridge, and they see, you can see the data is totally comparable between these two cartridges. And they have checked that there is no batch effect. This is again a T-SNE plot. So if you will see the cartridge 1 and cartridge 2, it looks like a replica of the cartridge. So the same data you will get it, and you can see all the sample tags were beautifully different. So this is one nice line, like sample tag 7, then the green one is sample tag 10. The same way, every sample is different from each and every time point. Any question? Yeah, it is similar. SIGING is same, even the interface is same. No, it is. No, okay. The problem is that this. Again, see, it's a business, they have to grow also. But the SIGING software is same interface, like add sample. You must be using a Flojo add sample, then right click and go for this. It's the same interface, everything is, all the plugins and everything is same, just because in single cell you have multiple plugins also for Bayesian plot, for differential gene expression analysis. So the plugins are different, that can be added more and more, but you have to install both the software, Flojo and SIGING. But I think if you have the license of Flojo, then SIGING is free of cost. So you can see this, there is no batch effect, and this data was checked between when cells. This is a 40-plex data. 40-plex means 40 antibody used in one experiment, and then it has been checked that what is how it looks like between resting T cell and then activated T cell. Totally different kind of islands you can see, and this RNA data is also correlated. Now they have checked again with the bone marrow extracted from these two mouse, and they wanted to check whether mouse 1 and 2 immune cell will be looked like the same, because if the mouse is different, same thing but in different cartridges, whether I will get the same data. Yes, you will have the same kind of population that you are getting from mouse 1, from mouse 2 also, and the gene is also kind of similar, correlated. So there is no batch effect, so it will give you the reliable and consistent results. So biological result, minimum sample bias, and you have ability to integrate large data without correction of the batch effect. That we generally do it in the Flojo. If you are using a large number of things, then you do it the batch effect, batch correction. So scanner is important. Why? Because it gives me the visual workflow analysis at each and every step. It will give me the estimated cost of the sequencing also. So before you pay, you know that how many cells you have loaded. So you know everything, and it will help me the troubleshooting also. So suppose that this is my experience, that somewhere I was working on some very difficult to handle cell, that is spheroid. So in a spheroid, you cannot count it into the normal Newborn chamber. So if you are going to load it, if you don't know that how many cells you have loaded, how you are going to say that you sequence these many cells. So if you have the scanner, you can check that okay, these many cells I have loaded, and I will go for this much of the depth for the sequencing. So it will give me the visual workflow, and it will tell me about the cell retention, troubleshooting and all. So if I'll ask a question, you see this blue, if this is a RNA analysis of, you can see these are some different islands of the cell. So how many population you can see in this blue circle? Kriti? How many population you can see? In this blue one, there will, if I'll say what this could be, 1 or 2 population. If I'm doing RNA analysis, so this could be different because they are at different different places. So now if I'll integrate my protein data, so then it will tell me no, this is not only 2 population, it is 3 population. So based off the integration of protein and gene data, and that way you always get a new biomarker, new cell types. So you can see here also, this is a CD4 island. It is giving me 2 islands of the CD4. Okay, on the basis of ABSEEK, this decent plot. And if I am using the CD45 RA also, then it will tell me RA positive and RA negative cell. Then if I am going to check this gene expression, then RA, just an example that we have did. So you will see that RA positive cells have different kind of a genes than RA negative cells, that we know. But that kind of data, this is just an example that you will have both protein and gene data together. This is one of the publication published in year 2020 when everybody is in lockdown. So there it is published in Cell, where they have shown that severe COVID-19 is marked by dysregulated myelin compartment. So if you have seen any of the COVID patient report, there is a high neutrophil. But in the severe, if somebody is COVID infected, so if I'll check through the flow cytometer, I'll know that neutrophil population will be 2 or 3 types maximum. But if you are doing the single cell, there they have used 40 antibodies and did the whole transcriptome. So the UMAP formation is not based on the ABSEEK part. It will make the population of the cell based on the gene as well as protein. So here it is showing there are not only 3 or 4 population of neutrophil. There is 9 different kind of neutrophils are there, and these neutrophils in the severe patient they express those genes which are basically expressed in case of a sepsis. So you know, if a patient is sepsis, all the immune system is at very bad condition. Multiple organ failure started and nothing works. So that's why so this kind of a data you will get it when you integrate both protein and gene. There it is unbiased. You don't know that what kind of a gene is going to predict, and this UMAP is based on the gene and protein data together. So because of this unsupervised algorithm, machine learning and all this software, they made this UMAP. And you can go through this paper. It's a very good paper. Later on they published another paper on this using our technology, absolutely, and they validated through the flow cytometer also this data, that this is the biomarker that is published. These were some highlights of this paper, but I'm not going in detail about that. So I told initially that we use this technology to resolve cellular heterogeneity. So generally, if a person is working on Tregs, you know that Tregs are important in multiple kind of things like acute infection and transplantation, cancer, somewhere on therapy and all. And how do we differentiate Tregs? It is naive Tregs or activated Tregs, or like based on this FOXP3 expression, CD25, 45RA, low, FOXP3 low or FOXP3 high. So this is the transition. So this HLDR and CD40, this generally gives me the 3 main Tregs population. So the experiment was designed in such a way, just think that if I am interested in Tregs only, so why should I check the other thing? So what I will do, I will enrich my population. So what they did, they took the PBMC, stained with the flow antibody, along with the ABSEEK also, and then they sorted. So we can use both the antibody. We can, if you wanted to, so there the picture of flow comes. So if you wanted to check only T-Rex. So how do? You have to sort it. So sorting requires flow machine. Then if my question is whether I can use both the antibodies together, AppSeq and this normal CD25 APC, yes, you can use it. There is a protocol, and on that basis you are going to stain, and then you can finally sort it. So after sorting, I know that my cell population is enriched only in T-Rex, okay, and then this population I'm going to take it for the cartridge. I'm going to load it onto the Rhapsody cartridge, and then I'll scan and isolate the RNA, then make the cDNA and the sequencing. So here is the panel that we have used it along with the flow panel. This is a panel of AppSeq, different markers. Now you can see this is known as monocole, which is an unsupervised algorithm, which has the ability to define transcriptional dynamics and trajectories that may occur over a time, for example, during cell differentiation and activation. So what does it do? So it will check, this is my protein expression, this is my gene expression. On that basis, it will tell me at that time of point, my naive cell looks like this. These are the different time points that my cell at different stages of my cell, and then when it will become the activated C cell. So on this basis, it is showing me the 9 population of the T-Rex. That is based on both protein and gene data, and so you will see that different kind of genes expressed at each and every population. And when you check the gene expression, same way you will have different gene for naive and effector cells. So somewhat if somebody is using Treg for their therapy, and you know that we should know that if at this stage of the cells we have to take, so that it will express those kind of a gene. So that kind of data you will get when you integrate both gene and protein data. And there are some papers, multiple papers on multiple things. So if you are interested, I can share that what is your area of research accordingly. So this is one of the example where they have used it for steroid. And these are some takeaways like single-cell multiomic approach enables new biological insight to discover distinct and novel cell population, and you can identify novel cell targets, and you can, best thing is you can identify what could be the pathogenesis at what step and how my data looks like when it is there, and you can go up to unprecedented depth and breadth because you don't know that what could be my novel cell types and what could be my gene profile. And later on, if you find out any rare population, you can further validate based on their marker. You can make another marker and you can further validate through the flow cytometer. So there are papers where people have used for ovarian cancer, even for TB also, that this kind of a population is there for identify differentiate between latent TB generally and the healthy control. What could be the thing that we can get it in that scenario. So these kind of data you will get it. So thank you. So if you have any question, I'm happy to discuss.
The session is open for questions.
Good morning, ma'am. Thank you for the insightful talk. Ma'am, my question is when you talk about the pooling of the samples, so won't be any issue with the mixing the samples and later on differentiating it? So, okay.
No, suppose I'm giving you an example. If you have pooled 6 samples, all the 6 samples will be sample typed. So what is the protocol? How do you do it? So generally with the flow cytometer, if you are going to stain your cell with a cocktail of antibody, what do? You took your cell, make it to single cell suspension, then you incubate with your cocktail of antibody for 30 minutes, 45 minutes depending upon your thing. So in that way, similar to that, this sample multiplexing kit is containing some vials where we put our sample, single cell, into that vial and we incubate it for 20 minutes. So that ensure that even at 20 minutes, it will go and bind to my population, then we wash it off, then we pool it. And the question is like, after the sequencing, how I will demultiplex our data? Whether I'll get the sample tag, suppose your sample tag 1 is for healthy control, 2 is for patient. So the same way I'll get it after the sequencing or not, and how it will happen. So we have a pipeline which is there is known as 7 bridges pipeline. So when you do the sequencing, you will have the FASTQ files. FASTQ files, you don't know that how many samples you have loaded. So once you have run that FASTQ files on these 7 bridges, you will put some command. You will say that I have used this sample multiplexing kit and it has these tags. Or if you have not put it also, then also it will give you that you have used 7 tags or 6 tags. In all those 6 tags, how many cells are there? And the question is that whether it will be on same or not, ratio will be the same. Whether if I have pooled it. So the pooling ratio, what we do, we generally pool it. We have a kind of a mathematical formula where we keep the pooling ratio 1. So in that ensure that each and every population we took the same number of cells. So if you are going to pool 4 samples, then 10,000 per sample. So I expect that after sequencing it should be 8,000 per cell, but generally it doesn't, whatever expectation is not like that. You will not get 8, somewhere you will get 6, somewhere you will get 7. So it's all depend upon the PCR and again the noise of the sequencer, noise of your work, so that way. But you can always demultiplex.
So now this pooling is the advantage of your technology, or it is the advantage in other also, like we can do the pooling in other technologies.
As per my knowledge, you can do it in other also, but here you can go up to 12. That I'm not sure whether there you can go up to 12 or not. Here, there I don't know, but I think there you can go up to 4 or 5. But here you can go up to 12 samples in one.
Okay. One last question now. Ma'am, where you talked about the 55 antibodies, so in that we can use it for the same samples or different samples?
See, 55 different vials of antibody. You take 2 microliter from each of the antibody, 2 microliter from each of the vial, make a cocktail, and then your pooled sample is, it could be 12, it could be 4, it could be 3, depending upon your choice. So you will take the antibody amount that you are going to take it from all those vials is only for 1 sample, but it is for 12 samples. So you can use it for further also. Suppose your project is on 64 sample, and for 64 sample you wanted to pull 6 samples per cartridge. So in that way you are going to pull 6 samples, and this kit will allow you to add 6 samples, but the amount of antibody you are going to use it for only for 1 sample. So the antibody you don't have to titrate like for antibody. Like if you are going to use it for 1 million cell, you can use it for up to 1 million cells. So, but because here we are going up to only 40,000 cells, so there it is only one sample antibody will be used for all the 6 samples. Yeah, I always prefer to make cocktail. So what we do, we took 55 antibody cocktail into one tube, and then we add the sample. So that sample is already tagged, washed. It could be 6, 7, 8 depending on that, and then we always do it like that. We always make a cocktail with the protocol. Always suggests that you have to first make the cocktail, and the cocktail is 2x. So then when you add the sample, it will become 1x. And we made 3 different kind of libraries. Suppose, because here I am talking about sample multiplexing, I am talking about AppSeq. So AppSeq library is also there because you have to check the protein. And if I am talking about whole transcriptome, there is another library of whole transcriptome. Now the question is, before sending to the sequencing, how do I ensure that my libraries are good? QC is passed. What is the QC criteria? So generally for the QC, if you have used the sequencing, so you check the qubit concentration, you do the nanodrop or anything, but here we do the qubit as well as the bioanalyzer. So bioanalyzer you have to check it, and that will suggest me that the peak should be at 600, it should be of this much concentration, then only you have to send it. So that we check at each and every step. There are multiple rounds of PCR that we have to do it for generation of these libraries. No, we provide only kits. And I am the person who nowadays very less than fax machine. Analysis. So analysis, I told you that it is through the CGEB software, and the 7 pages pipeline is free of cost for all the big users. So you have to tell that you are a big user, and you can further check them. It's a third party 7 pages pipeline. It's a cloud-based service. So we always write this mail, and you have to open the account. Once the account is open, you can upload it, and will always help from the data uploading of the 7 pages to the data analysis. Because if you will talk about single cell, that I am the person for that, you have to talk to me that how to upload the data, how to analyze it, and so on. Yeah, so yes. See, if you are not interested in protein, you already sorted your cell. There are two different kind of cells. One is your monocyte population, intermediate or classical. You want to sample. You sample type this non-classical, classical, and intermediate monocyte. You don't want to use APSI. So what do you want? You will have. You can say it's a single cell RNA, but here we are not having RNA because we are converting RNA into DNA. So everything is now it's in DNA form. You will get all the transcriptome data. There is no rate score. There is no ring quality that you are going to check it. Everything is through the qubit high sensitivity DNA kit, Barn-Leiser high sensitivity DNA kit. So you will check the product size based on the DNA content. So if I am saying that after the PCR the product size should be around 2000 base pair, so it should look like the 2000 base pair from. So I can show you the how the product look like. So that way you will check the QC. And after the suppose your question is like I don't want to use APSI. I wanted to use it only for sample multiplexing and whole transcriptome. Not interested in APSI because I know my APSI part. I know these are two different populations. So there you can do it. There our pipeline is like that. You can use only WTA, but it will not support without WTA or without G. If you will say I wanted to use only 55 antibody, forget about whole transcriptome or tablet. No, till now we don't have this provision that you can use only protein data. You have to use gene data also. But it will demultiplex at each and every time what could be my gene, what could be my protein, and that demultiplexing is done at 7 bridges, and you will have the files in CSV format, all the Excel file, and you have to transport it into the CGEB. And 7 bridges also give you overall thing that what could be my immune cell classification, what could be my cell, but it will not tell you the secondary analysis, that Wine plot or other things. So you can take this file. If you don't want to use CGEB, you want to use your own pipeline or our software, Syrup package, or there are n number of packages available in the market. You can use that also. Yes. Yeah. Yes. Yes, because you will have all the gene transcriptome data, all the gene data. So all the 20,000-30,000 gene data you will have it. So you will get differentially expressing. How do you check the differentially expressing? Generally, so there if you will do you can do mostly 1 to 10 gene at a time. Here you will get the data of 2,000, 3,000 gene at a time of single cell. Yes. But what do you want for? So housekeeping you will not interested. You will put a cutoff and then you will take that part only the differentially expressive. Yes. Yes. So there will be it will show on a plot, rank plot that these are my genes. Okay, it's a new plot or rank plot we call it. And then you have to select those genes which are not housekeeping, which are expressed at each and every cell. So I am going to select only differentially expressive. So generally with differentially expressing, if you have a very good depth, you will get more than 2,000 or 3,000 differentially expressed genes, and then with that you can do it. But But if you're interested in targeted panels, suppose, in targeted also you will get the same thing. Like you will have the same. The cost part you have to talk to. I'm not sure about, but I'm telling you it's not very costly. Instrument is not very costly. If you will go, you mostly work on VMCs or mouse. I don't know. Mouse not. Okay. So you can go for only for the express also. It's not very costly, but I'm telling you it's not very costly. It's very cheap. Kits are also like that. And the best part is that if you're going to use mouse, same we are having the mouse, whole transcriptome, onco panel, everything is there. And we have the sample multiplexing kit for mouse also. So you can put, I did some experiment at some place in Calcutta where they worked on the mouse sample and the melanoma also scale. So there also we have did it. So it's very easy. Yes. Yeah, sure. I'm happy to do it. Yeah. You can do it in lab.
Thank you so much, ma'am, for your insightful lecture. Now I would like to request Dr. Ved to please facilitate Dr. Ramsha. So now I would like to welcome Dr. Vinay Kumar Gupta from the Application Manager, BD Biosciences, New Delhi. Dr. Gupta has received his PhD from All India Institute of Medical Science, AIIMS, New Delhi in 2010 in Immunology of Tuberculosis. He joined BD Biosciences India as an application scientist in 2012 and has appreciable expertise in flow cytometry. Till date, he has trained more than 600 candidates in various programs, and currently he is working as an application manager and provides on-site technical support to flow users on conducting post-installation training, refresher training and troubleshooting of technical issues. He has participated as faculty in more than 25 workshops, conferences and till date including TCS and Indo-US workshops. Let us welcome Dr. Vinay to introduce us to the fascinating world of flow cytometry.
Thank you for your kind words. Good morning, everyone. And I want to thank Dr. Ved for providing us an opportunity to talk about this important cell analysis technology. So, biologists have been trying to understand how cells function. They are equally interested in understanding why and how do cells malfunction. Functions and malfunctions have been studied at various levels. Right from the organism level, we have gone down to the molecular level. but still we do not completely understand the specific targeted control on cellular events which govern their functions and wall functions. It has been realized the techniques, most common techniques to study cellular functions, they largely depend on life rates of thousands or millions of cells. For example, what you do in Western blotting, you will have your cell of interest, you set up the culture, you give some challenge, you harvest the culture, you prepare lysate, you run on gel, transfer on membrane, probe with a specific antibody, and you know all cells will not behave in the same fashion against a given stimulus. So there your actual information gets directed. What you get, that is average information. That is where flow cytometry is different. Here we are not interested in getting the average information. Rather we study one cell or one particle at a time. So there are two major systems for studying cell function, flow cytometry and image cytometry. So I'll focus on flow cytometry by which you can rapidly study many parameters. So it all started around 1950 and the first commercial flow cytometer came in 1967, and in the same year Heisenberg at Stanford coined the term FACS, which stands for fluorescence activated cell sorting. The basic definition is flow cytometry is a technology that simultaneously measures and then analyzes multiple physical characteristics of single particles. That is important. Usually cells. By nature, all flow cytometers are particulometers. We are biologists feeding cells, calling it cytometer. Usually cells, as they flow in a fluid stream through a beam of light, usually a laser. So basically in flow cytometry, we are interrogating cells with the help of laser light flowing cells. As a result, they emit some scatter patterns, some fluorescence patterns. We collect those patterns to drive a lot of information. On all flow cytometer, we get information about the relative size in terms of forward scatter. We also get information about relative granularity or internal complexity in terms of size scatter, which is attributed by various macromolecules, internal organelles. And we also get information about the relative fluorescence intensity. This much information we get from all flow cytometer. As you can see, it gives information about the relative cell size. That means if we run a heterogeneous cell suspension on a flow cytometer, then it will give us information, these are smallest size cells, these are medium size cells, these are largest size cells. If we really know the exact size of the cell, then probably flow cytometry was the right technology. Same way, when we run, let's say, human RBC live peripheral blood, it will show these are lymphocytes because their internal complexity of granularity is least. These are granulocytes because their internal complexity of granularity is highest. Same way, when we stain cells, the fluorescent label antibody, it shows these are dimly stained cells, these are brightly stained, so it's always relative. Now, how do we measure the forward scatter? So there is a cell, light comes from this direction, there is some bending of light at the edge. What we do? We place a detector in the direction of incident light in the forward direction, somewhere there. Forward scatter related to the cell surface area, and it is detected along axis of incident light in the forward direction. So basically, laser beam hitting as such. Cells are coming one after another like the obedient guys. And due to bending of light, there is a cone formation in the detector. So when a small size cell passes, it forms a small size cone. When a large size cell passes, it forms a large size cone. And their proportionate voltage pulse is generated. That is more important. Because in flow cytometry, all the signals, whether it's scatter or fluorescence, all the signals are measured in terms of their voltage pulse generation. We'll discuss in detail how voltage pulse is generated, but more height means more signal. Forward scatter is like if you have two different size objects, and if you throw a torchlight, you will see two shadows on the wall. Just by looking at the size of the shadow, you can comment which is smaller in size and which is bigger in size. That is forward scatter. When you throw light, it gets scattered in all other directions. So what we do? We place a detector at right angle at 90 degrees to the incident light that gives us information about cell granularity and complexity. It's like if you take water in a glass and you throw torchlight, light will be scattered in all the directions. Then you take some chalk powder, you add that glass, again throw same torchlight. So now it will scatter more light. It will be like a colloidal suspension, right? Each particle will scatter light. Same way, if the internal complexity or granularity of cell is more, it will scatter more light. You will get more value in your size scatter detector. That is why typical human RBC live peripheral blood picture look like this when we resolve on forward scatter, which gives information about relative cell size, versus side scatter, which gives information about internal complexity. If we look at this plot, now we realize monocytes are bigger than lymphocytes. Why? Because monocytes on the forward scatter are coming close to 100 channel position, whereas lymphocytes are coming close to 50 channel position. And this 50, 100, 150 is an arbitrary scale. It has nothing to do with the size of the lymphocyte. We know lymphocytes are least granular. Look at their SSC value, that is least. Neutrophils are highly granular, SSC value is also high. and monocytes are intermediate in terms of granularity, so their FSC value is intermediate. This slide shows the range of particles which you can analyze by flow cytometry. On most of the flow cytometer, you can work well within this range. To be more precise, 0.5 to 70 microsize particles can be studied on most of the flow cytometer. But if you work on something which is close to 100 microns, then that's a challenge. Similarly, in some instruments, there is a small particle detection option where you can detect particle up to 200 nanometer. And recently, BD has launched a platform FACT Symphony A1, which is capable of detecting as small as 90 nanometer particles. So after scatter comes fluorescence. Basically, it's a phenomenon where the molecule emits light of lower wavelength, and within nanoseconds, they emit light of higher wavelength. As you can see in this slide, antibody is tagged with FITC, fluorescent isothiocide molecule. This molecule has property when you throw light in the blue region that for 18 nanometer light, it absorbs energy. So initially electrons are at ground state. After absorbing energy, they go to the excited state. But they cannot remain in excited state. Within 1 to 10 nanoseconds, they will come back to the ground state. While coming, they emit light in the form of photon. And in this transition, there is some loss of energy in the form of heat and vibration. And as we know, energy has inverse relation with wavelength. As a result, the emitted photon will have higher wavelength than the incident light. So these fluorochrome molecules, they absorb light over a range of wavelengths. And at one particular wavelength, they will absorb maximum light. We call that absorption maxima. Similarly, they will emit light over a range of wavelengths. And at one particular wavelength, they will emit maximum light. We call that emission maxima. The wavelength difference between emission maxima and absorption maxima is known as Stokes shift. The fluorescent intensity is proportional to the antibody binding site. As you can see, there is a cell which has just one receptor, and there is a cell which has a couple of receptors. When you stain these cells with specific antibody, this will appear as dimly stained, this will appear as brightly stained, because fluorescent intensity is proportional to the number of antibody binding sites, and that is how their verticals will look like. Now, what are the basic components of flow cytometer? So, all flow cytometer will have these basic components: fluidics, optics, and electronics. Fluidics, first you need to resuspend yourself in some liquid media buffer. Then there should be something to carry, transport yourself to the point of laser electrolysis, right? That is where fluidics comes in, to maintain a single file flow system. Optics, there are two types of optics in flow cytometry: excitation optics and collection optics. First you need to excite the fluorochrome with the help of laser light. Then whatever signal is generated, you need to collect with the help of collection optics. And the third component is the electronics, to convert the photonic signal to the proportionate digital signal. Okay. Overview of flow cytometry process. Cells are coming one after another like Ovidian guide with the help of fluidics, the single cycle system. You interrogate your cells with the help of laser light, and immediately after interrogation, in the direction of incident light, in the forward direction, you collect your forward scatter signal. Rest all the signals you collect in 90 degrees, the signals are filtered, then sent to respective detectors. Finally, all the signals are sent to the electronics to generate display and statistics. That's the part of fluidics, excitation optics, collection optics, and electronics. So what happens in a flow cytometer? Cells are moving in a single file flow system with the help of fluidics. There is laser interrogation point. After interrogation, they emit some scatter patterns, some fluorescent pattern that is collected, filtered, measured. And with the help of electronics, that is converted to digital values that are stored in a file, and later we use software to analyze and interpret our results. Now let's look at these components in detail. Fluidics. This is a typical fluidics diagram of a flow cytometer. So there is air pump. Air is sucked in, it is filtered. There is 0.22 micron filter. Then there is something known as sheath. The term sheath means what? Covering, right? So in flow cytometry, we use a buffer. We commonly call that buffer sheath fluid because that buffer forms covering around our sample. It is basically nothing but 0.22 micron filter, 1x PBS with some additives. Okay. With the help of air pump, we pressurize the sheath tank. Sheath moves out. There is a filter. It gets filtered. Then it goes to flow cell, which is known as heart of the flow cytometer. Then it goes to waste. What we do? We prepare our sample, we load our sample, and we also pressurize our sample tube. As a result, sample moves up, it joins the path of the flowing sheath, and somewhere at the stem of the flow cell, lasers are setting, where all the interrogations are happening, generated signals are collected. Finally, everything is taken to waste. So we are pressurizing sheath tank, we are also pressurizing the sample tube. These pressures are not same, they are different. Sample pressure is slightly higher than what you apply on the sheath tank. And there is a sample regulator. Basically, the rate by which sample is entering in the flow cell, that can be controlled by the sample regulator. You have options of running sample at low flow rate, medium flow rate, high flow rate. In some instrument it is software driven, and some instrument you will see separate knobs to adjust the flow rate. But in all flow cytometer, at all the flow rates, sample pressure is always higher than what is applied on the sheet tip. So sample is running, sheet is running continuously. What you do in the center of flowing sheet, you introduce your sample in such a way that their core becomes focused along the central axis. That is known as hydrodynamic focusing. So when you introduce your well in the center of flowing sheet squeezes your sample from all the directions. There is equal pressure on your sample from all directions. It cannot move to right or left. It only moves in the forward direction, and it forms a virtual capillary in the center of flowing sheet. As such, there is no capillary. It just forms a virtual capillary. Diameter of this capillary is so narrow it allows only one cell to pass at a time. And this inverted funnel design of the flow cell, it helps in attaining the laminar flow. There are two types of flow. One is laminar, other is turbulent. In case of turbulent flow, two fluids cannot flow simultaneously parallelly without getting mixed. But in case of laminar flow, two fluids can flow simultaneously parallelly without getting mixed. That further depends on a factor known as Reynolds number. If the value of Reynolds number is less than 2300, you attain the laminar flow. If it exceeds 2300, that becomes turbulent. That further depends on many factors. What is the density of the fluid, what is the viscosity of the fluid, what is the core diameter, what is the speed. But essentially, in all flow cytometers, we always attain the laminar flow. And this is how picture look like when you run your sample at low differential pressure, and this is how it look like when you sample at high differential pressure. Can you see any difference between these two pictures? So what happens at high differential pressure? See, so I told you there are three options running sample: low flow rate, medium, high flow rate. Let's assume at low flow rate, sheet pressure is 5 psi, and when you are sample at low flow rate, you are applying just 5.2 psi. That means pressure difference is good enough. When you move from low flow rate to high flow rate, this will remain 5 psi only, maybe that will become 5.8. So at high flow rate, at high differential pressure, we are applying more pressure on our sample tube. As you can see here, we are pushing 5 times more sample. Look at the diameter of the virtual capillary. We are increasing the diameter of the virtual capillary because we are pushing 5 times more sample. The laser beam is sitting as such in the center. So when you are running sample at low differential pressure, laser beam is sitting as such, cells are passing exactly through the center of the laser beam. But since you have increased the diameter of the virtual capillary, laser is still sitting as such. But now there is a chance either cell will pass exactly through center or slightly through this side or other side. As a result, our resolution is little bit compromised when we run sample at high differential pressure. Because the event which is crossing the beam through this side or other side will not reveal as high number of photons as received by the event which is crossing the beam through the center. So, how do we take call when to run sample at low flow rate, when to run sample at high flow rate? Most of the time we run sample at medium or high flow rate. Depends on the assay, what assay we are doing. For assays like DNA cell again, we run sample at low flow rate. For immunophenotyping, like studying 3, 4, 8 like stuff, it doesn't matter whether you sample at low flow rate or high flow rate. Optics. So optics has two components: excitation optics and collection optics. Lasers are part of the excitation optics. Lasers are mounted at different places on the optical bench. So we use fiber optical cable to route the laser light to the point of interrogation. And collection optics, after interrogation, whatever signal is generated that is collected by the fiber optical cable, and the collected signal is taken to the respective emission blocks where we have photomultiplier tube which will convert photonic signal to the electronic signal. As you can see, blue laser, red laser, signal is taken out to the fiber optical cable. We use route the laser light to the point of interrogation, and after interrogation, whatever signal is generated, that will be collected by the fiber optical cable and that will be taken to the respective emission blocks. These are some of the commonly used excitation source laser lines. Blue laser, which is 480 nanometer line, we call primary laser, which you see in all flow cytometer. Second most common laser is the 630 or 640 nanometer red laser line. Then depending on your assay, you can choose laser line of interest. Like you are working on DAPI. DAPI is a DNA magnetite. And if your intention is to work with the DAPI state cell, then your flow cytometer should have the 355 nanometer laser line. Only that can excite the DAPI. So depending on your interest, your application, you can choose flow cytometer. Your flow cytometer, which is basic flow cytometer, which has two lasers, blue laser, red laser, and that is 4 color, to all the way 9 laser, 50 parameter, 48 color flow cytometer. So in flow cytometry, we commonly use two types of detector: light sensitive diode, we call them photodiode, and the photomultiplier tube. So for forward scatter, we use photodiode, and for rest everything, we use photomultiplier tube. Photodiodes extensive use for high signal like forward cut signal. So after interrogation in the forward direction we collect the forward cut signal. Then we collect maximum part of the signal. So we use less sensitive, less expensive devices which are photodiode, and rest all the signals we collect in 90 degree. There we take only some part of the signal. So we use much sensitive, much expensive devices which are photomultiplicative, which have high signal to noise ratio. And in flow cytometry we commonly use these optical which will have certain cut-on and cut-off wavelengths. As you can see, long pass LP500, it will allow longer than whatever is mentioned nanometer signal to pass. So this will allow more than 500 nanometer wavelength signal to pass. Anything which is less than 500 nanometer, it will simply block. It will not allow to pass. Then there is a short pass filter, SP500. This will allow anything which is less than 500 nanometer wavelength. Anything which is greater than 500, it will simply block. Then there is a band pass. This says BP500/50. So in nomenclature, band pass filters are always written like that: large number / small number. So this will allow a bandwidth of 50 nanometer, a bandwidth of 50 nanometer across 500, 25 this side, 25 this side. Any signal which is in the range of 475 to 525, it will allow to pass. Anything which is less than 475 or greater than 525, it will simply block. Then there is one more filter which is known as dichroic filter or mirror. The property of dichroic is, if you place a dichroic 45 degree angle to the incident light, then it will transmit certain signal and it will reflect certain signal. If we place long pass 500 dichroic at 45 degree angle to the incident light, then it will transmit more than 500 nanometer wavelength signal and it will reflect less than 500 nanometer wavelength signal at 45 degree. That means 90 degree in respect to the incident light. If we place SP500 dichroic at 45 degree angle to the incident light, then it will transmit less than 500 nanometer wavelength signal and it will reflect more than 500 nanometer wavelength signal. This is the classical way of arranging detector known as herringbone pattern. So as you can see, immediately after interrogation, in the forward direction you collect your reference signal, rest all the signals you collect at 90 degree, then signal pass through a series of dichroic. At each dichroic level, some part of the signal is transmitted, some is reflected, transmitted, reflected. So in this kind of arrangement, there is huge loss of signal at each detector level. So in modern flow cytometer, we do not follow this kind of arrangement of detectors. In modern flow cytometer, we prefer reflectance over transmittance. As you can see in case of transmittance, if at the first detector level you have 80% signal, by the time you reach 6 detector, there is huge loss of signal. In case of reflex, loss is minimal. This is how your detector look like. We call them octagon, trigon, based on the number of PMD it has. PMD is our detector. This is a blue laser octagon. The A, B, C, D, E, F, G, H are photomultiplier tube. And before the PMD, we are using particular type of filter. This says 695/40, 575/26. Basically, these are band pass filter. In the center, we are using long pass dichroic. So basically, what are we doing here? We are first collecting longer wavelength signal, and at the end we are collecting shorter wavelength signal. Advantage of doing that, we are collecting longer wavelength signal first. That means longest wavelength which has least energy or dimmest signal. So we are processing dimmest signal first. At the end we are processing our strongest signal. If we go for short pass arrangement instead of long pass, then our sequence of detection will be reversed. Then in the beginning we will be capturing signal which is strongest, at the end we will be capturing signal which is dimmest. By the time we reach the last detector, the weakest will die. The loss is minimal, but still some loss is there, right? So better to process the dimmest signal in the beginning. This slide shows the absorption spectra or excitation spectra of some of the fluorochrome which are excited with blue and red laser. So it is important to know what is the absorption spectra or excitation spectra of a fluorochrome before you work on a flow cytometer. Let's assume you have a flow cytometer which has a blue laser, just 480 nanometer line. There is no red laser in your flow cytometer. On that flow cytometer, you want to use anti-CD3 antibody tagged with APC. APC is a fluorochrome, yellow phycocyanin. Now look at the absorption or excitation spectra of APC. Hit by the blue laser, then it goes like this. That means you cannot excite APC with the help of blue laser. There has to be something in this range. Now look at the excitation spectra of FITC. FITC absorption maxima lies at 490 nanometer and we are using blue laser which is 488, which is nearly perfect to excite FITC maximally. Look at the critical spectra of PE, this dotted line. Hit by the blue laser, then there is a dip. It goes like this. That means PE can be excited with blue laser 488 as you can see in the slide, as well as with yellow-green laser which is 561 nanometer wavelength. 561 will come somewhere there and that will give you better excitation of PE, better than blue. In the instrument where we have yellow-green laser, we prefer to read PE in yellow-green rather than in blue. Okay. So when someone asks can you use this fancy fluorochrome labeled antibody in your instrument, you already know what laser lines are there in your instrument. That's not going to change on day-to-day basis. Just look at the technical data sheet of that product to understand what is the absorption spectra of that fluorochrome. Will I be able to excite the fluorochrome by the laser line fitting in my instrument? Yes, you checked you have the right excitation source, right laser line to excite that fluorochrome. Still, you cannot use that antibody. You also need to check what is the emission spectra of that fluorochrome. Do I have right set of detector to capture the generated emission spectra? That is where knowledge of emission spectra will help. FITC was excited with blue laser 488. Now it is emitting signal close to 530 nanometer. PE was also excited with blue. It is emitting close to 570 nanometer. Before you work on a flow cytometer, you must know the excitation as well as emission spectra of the fluorochrome tagged antibody which I am going to use. And this is how picture looks like when you put multiple fluorescent labeled antibody in the same tube. Their emission spectra are overlapping. Detector gets confused what signal is coming from where. The third component is the electronics, which will convert your analog signal to the proportional digital signal. Okay. This is your laser beam. Cell is just entering the laser beam, moving in this direction. So a small part of the cell will be interrogated. So fewer photons will be emitted. These fewer photons will reach the PMT. PMT will convert proportionate fewer electrons. If you circulate those electrons, you can measure reading. If you make a voltage versus time pulse, you will see some deflection in the voltmeter. When your event of interest in the center, that time maximum number of photons will reach PMT. PMT will convert maximum number of electrons, and you will see high reading in the voltmeter. When it goes away, you will see a drop. That's how voltage pulse is formed. So we measure height and area of the pulse. Height is maximum digitized value and area is sum of all the height. And we calculate width, which is known as, which is equal to the time of flight. The time taken by the event in crossing the laser beam. That is pulse width. Width parameter helps in excluding the aggregates. See, blood is a perfect example of single cell suspension. We don't have aggregates in blood. But when we work on a PBMC or some cell lines, and if we have a look of vibration under a microscope, we'll never get 100% single cell suspension. There will be aggregate, 1 or 2 dimer cells, gain, immunity, cell resticking. And in flow cytometry, we are not interested in getting the average information, right? We are interested in studying one particle at a time. So if there are aggregates, we need to exclude them from our analysis. Width parameter helps in excluding. I told you the width is proportional to the time of flight, the time taken by the event in crossing the beam. If two events are sticking together, if they will move across laser beam, that will take more time. So pulse width will increase. So you can exclude cells which are having higher pulse, higher width as aggregates. We'll show you in the exercise. So when we run a heterogeneous cell suspension which has lymphocyte, monocyte, neutrophil, all we see is this kind of plot, lympho, mono, neutro. We don't see pulses. That's the job of the instrument electronics to take care. But how do we get this plot? Photon is converted to an electron. This becomes a current signal. Current is amplified, digitized, and converted from a continuous number to a digital number by a DC converter. Digital number is sent to the computer, and finally, after processing, we generate picture like this. Lymphocytes are small in size. Look at their FSC pulse height. Small pulse height. Neutrophils are much larger in size. Look at their FSC pulse height. I told you more height means more signal. Lymphocytes are least granular, right? They are agranular. Look at their FSC pulse height. Neutrophils highly granular. Look at their FSC pulse height. But we don't see these pulses. All we see is this kind of plot. So photon comes in, you apply voltage across your PMT, signal is out, pulse is digitized, analog signal is converted digital signal, digital data is sent to the memory, and finally after processing you generate display and statistics. How do you display flow cytometry data? Most commonly used plot. This plot is known as histogram plot. Histogram is basically a frequency distribution curve, and histogram is a single parametric plot. You can have one parameter of interest on x-axis. On y-axis by default it shows number of cells, count, event. And these kind of plots are known as dot plots. Why dot plot? Because each dot represents data generated from a single cell. And dot plot can be divided in 4 quadrants. It's a biparametric plot. You can have one parameter on x-axis, one parameter on y-axis. So we consider these dots positive for the parameter which is on x-axis but negative for the parameter which is on y-axis. Similarly, these dots are considered positive for the parameter which is on y-axis but negative for the parameter which is on x-axis. And this quadrant is considered as double positive, positive for both. And this quadrant is considered as negative for both, double negative. So basically, this quadrant is showing just the level of autofluorescence. These are various ways to represent flow cytometry data. Dot plot, color density equal to plot where data is shown in the form of percentage gains. Just a matter of representation, your stat is not going to change, it will remain same. How do we store flow cytometry data? Data is stored in the form of LMD file, which is nothing but list mode data file. We are acquiring cells one by one. Let's say we are acquiring 10,000 cells. First cell was acquired and that gave, let's say, 638 value for the fluorescence 1, 840 value for fluorescence 2. We mark the position. 638 for fluorescence 1, 840 for fluorescence 2. Then came second event that gave 245 value for fluorescence 1, 85 for fluorescence 2. Again we mark the position 245, 85. Like that listing is done for all the 10,000 events for all the parameters. Any software which can read this kind of file should be able to create this kind of plot. If it's a matter of 4 or 5 events, then we can also make dot plot by looking at this file. But we deal with thousands, millions, so we take help of software to read this kind of file to get this kind of plot. There is a term known as gating. So what is gating? Gate is selection of cells on the basis of predefined properties. Like we are interested in studying lymphocytes, what we do, we take blood and we stain blood with antibody which are specific to lymphocyte like T lymphocyte, B lymphocyte. And when we analyze our data, we draw a region around population of interest. And we get one more plot which is gated on the drawn region. So regions are drawn, gates are applied. Okay. It's a dot plot, so we can have one parameter of a T cell on the x-axis, parameter of B cell on y-axis, and dot plot has 4 quadrants. So now we know 74% are T cells, 9% are B cell. What are these cells? NK cells. Because plot is gated on lymphocyte, that means it cannot show anything other than lymphocyte. If you stain this paper with antibody which are specific to NK cell like 360, 356, you can always cross check. So flow cytometry we see 2 types of scale, linear scale and log scale. So when do you have linear scale and when do you have log scale? When your data range is small, you go for linear scale, like DNA cell cycle we study. We are looking at 2-fold difference in the density G0, G1, G2, and we go for linear scale. Scatter, generally lymphoma, neutro, we use linear scale. Lymphocyte size is 8 to 10 micron. Monocyte, neutrophils are bigger, but how many fold? 1, 2, 3 fold, or more than that? For immunofluorescence, like CD348, where your negative and positive are 1000 fold apart. Their data range is large, so we go for the log scale. Because if we go for linear scale, then either we will be able to show negative or positive, not both on the plot. And in flow cytometry, everything is relative. Your positive is positive in respect to negative. You have to show negative also on the plot. How many events to be acquired? There is no upper limit. Try to acquire as high as you can. If your population of interest is 2% or above, then acquire at least 10,000 events to make your stat significant. If the population of interest is less than 2% and greater than 0.4, acquire at least 50,000 cells. If you want even more rare cells like peripheral blood genetic cells, myelogenetic cells, lymphogenetic cells, their percentage in peripheral blood is less than 0.1. In that case, you need to acquire at least 0.2 million events to make your stat significant. What is so unique about flow cytometry as a tool for cell analysis? Multiparametric, we get information of large number of cells rapidly, and we get information at a single cell level, allows physical isolation of cell of interest. So there are 2 types of flow cytometer across industry. Some are cell analyzers and some are cell sorters. There are flow cytometers where you can physically separate out the cells which you have just analyzed. Those are cell sorters. All cell sorters are cell analyzers. First you need to analyze, then only you can sort. Is there any limitation of flow cytometry technology? No limitation. Size limitation is there, right? I told you already. 0.5 to 17 microsize. Not ideal localization of the antigen. You are working on some intercellular marker. So if you add antibody that cannot go inside cell, right? You will have to permeabilize the cell membrane by treating some detergent. Once you have permeabilized the cell membrane, and whether it's going inside cytoplasm, inside nucleus, you have no control. You cannot comment. Right? Because we are not observing anything like microscopy. So not ideal for cellular localization of the antigen. Size limitation is there, and if you work on some tissue sample, lung, liver, kidney, whatever, if you cannot get single cell filtration out of that tissue sample even after acute or displaced treatment, if you cannot get monodispersed single cellular suspension, you cannot do flow cytometry. That is the limitation. So at BD Biosciences, we design, manufacture flow cytometers, monoclonal antibody, kit for analysis, reagent for life research, and the in vitro diagnostic reagent. So this is a typical workflow. So sample preparation and data analysis workflow. So most of the time either we work with the peripheral blood, or we work with the PBMC, or with the cell line, or with the crab with the cells. The cells needs to be stained with a specific antibody. Either it could be direct staining or it could be the indirect staining. Once you have stained cell after following a proper SOP, then you need to set up your instrument. You need to check the performance of the instrument whether it is working fine, and then you need to acquire your sample. After processing, when you analyze your result, you can get your population of interest and you can get the stat, whatever signal you are interested in. Basically, what's happening in flow cytometer? We have stained cells with specific antibody which are temperature local. When those cells are moving across the laser beam with the help of laser light, the fluorochemicals are excited. So these emitted photons cause a photodetector to produce electric current. This current is small in magnitude. For the longer term processing, it is converted to large magnitude, but they are amplified. So during processing, there are single processors which will process the pulse, where numerical values are assigned to each pulse in terms of height, area, width. Those values are stored in a file. Later we use a software to read that file to create the plots. Now I will play a 5-minute video to summarize whatever I have covered. There is no audio. Audio is not coming. This has audio. It has.
How to avoid this fault result?
Say again, your question.
During IDA or iron deficiency anemia, the MCV size of RBC will be decreased, and machine will read RBC as a platelets. How to avoid these fault results?
Machine will, okay, so you're saying that RBC size will reduce, so it will fall in the platelet result.
It will be read as a platelet, and it will be show a fault result. How to avoid this fault result by flow
Okay,
cytometry?
so if on the basis of scatter, if it is difficult to separate, segregate those cells, then you can use a specific marker either for platelet or for the RBC to separate out your cells.
But can we modify in the future to add some specific marker along with size that we can detect specific mark in flow cytometry?
Yeah, that's what I'm saying. You can use for platelet, you can use like 41, 61, 42A, or for platelet, glycophorin A. For RBC, you can use 235A. Specific marker to identify either platelet to separate out or your RBC.
Based on that it means that maybe specificity of flow cytometry will decrease if we use like that?
No. If your scatter is same on the basis of scatter SLC profile, if it is really difficult to separate those cells, then you will have to use some specific marker to separate one of the population.
Okay, thanks.
And then you can back it.
Particles or cells from 0.2 to 50 micrometers in size are suitable for flow cytometric analysis. On some cytometers, larger particles can be analyzed. A cell is flow cytometry. If we break the term into three parts, flow implies fluid. Cyto means cell, and metry means measurement. So flow cytometry is a process of performing measurements on cells or particles that are in liquid suspension. It is a technology that simultaneously measures and analyzes multiple characteristics of single cells or particles as they pass through a beam of light. With flow cytometry, researchers can perform many studies, and clinicians can make diagnoses to help treat patients. For example, researchers can study how stem cells mature and differentiate into other cell types. Clinicians can determine the percentages of CD4 positive T helper cells to monitor progression of HIV/AIDS. Flow cytometry is a powerful technology for characterizing and analyzing particles of interest, such as cells, chromosomes, bacteria, and beads. All cytometer samples must be in liquid suspension. Cells from solid tissue must be disaggregated before analysis. Typically, particles or cells from 0.2 to 50 micrometers in size are suitable for flow cytometric analysis. On some cytometers, larger particles can be analyzed. A cell's or particle's relative size, internal complexity, and fluorescence intensity can be measured by flow cytometry. A flow cytometer is composed of three main systems: fluidics, optics, and electronics. The three subsystems work together to simultaneously measure multiple physical characteristics of single particles as they move in a fluid stream through a beam of light. The fluidics subsystem brings the particles of interest to the interrogation point, where they interact with the excitation sources. The optics subsystem provides the excitation sources and the components to collect light signals and route them to the appropriate detectors. The electronics subsystem converts light signals to equivalent electronic signals. The fluidics subsystem transports particles in a fluidic stream to the laser beam for interrogation. The portion of the fluid stream where particles are located is called the sample core. The point at which particles meet the laser beam is the interrogation point. Incident laser light is scattered and fluorescence is emitted as particles pass through the interrogation point. The optics subsystem consists of excitation and collection components. The excitation components include lasers, beam-shaping prisms, and lenses to route the laser beams to the sample core. When particles pass through the laser interrogation points, they scatter light. Any fluorescent molecules present on the particle fluoresce. Appropriately positioned lenses collect scattered and fluorescent light. The collection components steer the scattered and fluorescent light to the appropriate detectors. The detectors in the electronic subsystem convert detected light into proportional electronic signals. Electronic signals are then digitized, further processed, and sent to the computer. Data for each cell or particle is stored in the computer and plotted in various ways for further analysis.
Thank you, sir. I really enjoyed the entire presentation. Meanwhile, I have a question there are some substances which naturally produce fluorescence. Now, how we are going, if I give an example, reduced pyridine nucleotides, NADPH, or oxidized flavins like FAD, they produce naturally fluorescence. How we are going to measure or determine the amount in the flow cytometry? Yes, we are producing naturally fluorescence. Are we going to stain through some fluorophore, or how we are going to measure them? I'm going to say that I'm going to measure these substances which I mentioned, like NADPH and FAD. Or I said reduced pyridine nucleotides or oxidized flavins. I'm going to measure these, but on the other side, they are producing naturally fluorescence. Yes, these are autofluorescent substance. Yes. Sir, still you insist that we should stay in the true fluorophobe? Okay, thank you, sir. Oh. Yes, I think that will be nice. Yeah. Thank you, sir.
Thank you, sir. Yes, sir. Please, 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 sir.
Basically, so in 1967, there is another word called the Dutch flax, the fluorescent activated cell sorting. It was not sorting. When blood sampling, we could separate out lympho, mono, nucleo on the plot, then he called sorting. That's not sorting. Nowadays, sorting is physically separate or isolation of cells. Fact is British trade name, so we use facts even for our analysis. Like facts were, which is an analyzer, we use facts for it. Should we use recently always recently when the Purdue University there was a debate on this. We should drop this facts.
Thank you, Dr. Vinay, for such an informative talk. I hope this must have given a glimpse of highly advanced techniques being used in biological research. Let's stop for a brief break. We invite you all to tea at the faculty lounge. The next session will begin after 15 minutes, and before that I would like to invite Dr. Ved Prakash Trivedi to please facilitate Dr. Vinay. Thank you. Manager in Bio-Rad Laboratories, developing a successful product roadmap, market research, and providing solutions to research institutes and R&D companies. Please, sir.
Thank you. Good morning. I hope I'm audible. This is the most thing you listen for the last 2 years for the online meetings. So, welcome. I hope you had good, nice refreshments upstairs. So, I'll be talking about immunoassays, more specifically how sensitive they are for specific detection of antibodies in biological samples. Before I start the proceedings, I'll just brief what we are about in terms of Bio-Rad. Bio-Rad actually is a 75-year-old company, started in 1952 in San Francisco, Hercules, California. They started off with husband and wife, and it has currently it has more than revenues of 2.56 billion, more than 9,000 employees worldwide, catering 150,000 customers across India. Different products, we have around 10,000 products over, and we have a lot of spread in terms of the R&D as well. We have presence in Americas, Europe, we have R&D centers in Europe, manufacturing centers in Asia Pacific, Singapore, China, across Europe. To give you a gist of what Bio-Rad is and how spread across Bio-Rad is on a global scale. Now, for the products which we have, so we have basically 3 arms of our product lines. I come from the life science, along with me Dr. Prashant. There is also a group called clinical diagnostic. So basically, whatever HbA1c testing which you are doing in the lab in Metropolis, SRL, it is done on a chromatography system, and we are the market test in that. So we have multiple products for life science as well as clinical diagnostic. Today we are here to talk about one part of the product in life science, that is immunology, and how solutions which we have across. So just to give you a brief on the diagnostic segment across various segments of whether it AIDS, HIV, newborn screening, we have products, controls, quality controls spanning across the entire diagnostic portfolio. Coming to life science, what we offer in terms of the multi-omics solutions from Bio-Rad, starting from your gene transfer, the real-time PCR, so we have the 3rd generation of droplet digital PCR. Then we are talking about multiplexing, bead-based assay, chromatography systems. Bio-Rad is known for electrophoresis imaging platforms and those sort of stuff. Even we have cell sorter, flow cytometer, which is there. So coming to the topic which we are talking about, the immunoassays. Just to give you a gist on what basically immunoassay is. Most of you are aware, but just to give you a gist to start off the proceedings. Now, immunoassays play a very critical role in bioanalytical settings, more specifically towards clinical diagnostic in terms of determining what are the interleukin cytokines which are hampering in terms of the patient profile, whether in terms of biopharma industry, whether the vaccines which are developed, what are the immunogenicity during the development. They do all the mouse and rat trials, environmental monitoring, food testing. So there are various aspects of immunoassay. Again, precise early detection of analytes is essential requirement for any application. It is even more critical for diagnosing, monitoring, managing patient health. Now, couple of examples, especially in the last couple of years, we are into the COVID phase. When COVID started off, you heard about cytokine release storm, right? And there are few of the cytokines which were responsible for those storms. And these were detected on a multiplex because they were large amount of patients as well as samples. So these were detected on a XNAB, that is bioplex based system, wherein the IL-6 and the IL-10 which were responsible for those, and the patient monitoring and the treatment can be changed accordingly. So early detection helps monitoring the patients and also deciding what are the treatment post that. Also considering the prominent role of immunoassays which play in clinical decision making, as I mentioned, and having in terms of the indispensable healthcare settings. Types of immunoassay, just a broad perspective on various types. We have the age-old radio immunoassay. People are trying to move away because of the ill effects or the health hazards containing agents used. We have the counting immunoassay, enzyme immunoassay or ELISA, which is most commonly known and used across board. Then we have the flow immunoassay and chemiluminescence immunoassay. So these are the broader perspectives. We have seen lately that label-free immunoassays are also being discussed quite regularly of late. So what does Bio-Rad offer in this space of immunoassay? So Bio-Rad offers something called the XMAP technology. Now what is this XMAP? XMAP is multiple analyte profiling and X stands for the unknown. Now what is this about? This is a fluorescence bead-based technology developed by Luminex. So it's basically a technology developed by Luminex and we are the OEM. Now what this does is basically instead of in ELISA you do a single target on a single plate, right? So instead of that we have the options of monitoring 100 different cytokines in a single well, 100 or up to 500 depending upon whatever the instrument you are procuring. It is a powerful and alternative tool to your traditional ELISA. And how this is different, you can see from on the image on the left side, basically a planar surface. You have your capture antibody, basically detection. You have in between you have your target and your detection antibody. Basically, you have just sandwich immunoassay. But on the right, how the Luminex platform or the Bio-Rad platform works is instead of the ELISA plate, the capture antibody is immobilized on a bead. That is a magnetic bead. Okay, and these are basically how this works. I just show you the subsequent slide. Then the bead, basically the capture antibody captures your antigen. Then your subsequent sandwich immunoassay, the process works out for detecting those specific antibodies. Very similar to ELISA, but the assays is based on the bead suspension instead of plate. Now, if you see what are the benefits of doing multiplexing, basically from that perspective, you have increased amount of data coming in of each well. Rather than in terms of, as I mentioned, instead of doing just one target on one well, you can do up to 100. We have off-the-shelf kits of having 48 different cytokines in a single well, and you can generate a vast amount of data just from that one sample. Easier error proof, basically in terms of the software which is there. You have discrete sample volume. Now, if you compare with ELISA, traditionally you use around 50 to 100 microliter, but if the sample required for this is just 12.5 microliter. If you are working on any animal models, sample volumes are very difficult to get. So if you do consider that aspect, just 12.5 microliter for either a 48 or a 27 plex, depending on what targets you are looking at, is huge. And if you are looking at duplicates, you might require 25 microliter. We have also applications in terms of eye research. We are monitoring the tears, the cytokines in the tears, where people, scientists have even done analysis using 6 to 7 microliters. We recommend 12.5, but once they have got hold of the instrument, they have done even 6 to 7 microliters of sample. Reduce reagents, reduce time. These are the few of the benefits which comes across for the benefits of multiplex. Now, as I mentioned, the key aspect is limited sample volume. Now, if you are working in animal models, 1.5 ml is the whole blood available in the mouse. Okay, again 2 to 3 mm of ELT. So, when you compare sample volumes, yes, this becomes a very critical aspect. 12.5 ml of sample, which sometimes becomes difficult in your day-to-day ELISA protocols per se. And how do we compare from your traditional ELISA? If you are looking at, for example, 48 different targets or cytokines, you need to do a 48 different ELISA plates. The data point would be the same because 48 into your number of samples, that is KT. But in the Luminex or the Bio-Rad platform, it will run just one target or one plate which has all these 48. And the runtime basically is around 4 to 5 hours. And the read time on the instrument is just 45 minutes after your protocol is built up. On the last column, basically you can see just 12.5 vectors of the sample, which is needed for getting this amount of data. And after me, Dr. Prashant is there just to show how the vast amount of data can be interpreted using this platform. Now, the beauty of bioplex system is the bead, because bead is where you differentiate different targets, and how it has been done is this bead, this magnetic bead is immersed into dyes, red and infrared, at different proportions, and that becomes the crux of the story, because every bead has its unique spectral identification. For example, there are bioplex 200 can analyze 100 different targets. Now, what are these 100 different targets? 100 different targets are nothing but 100 different beads. 100 different beads which has a unique spectral identification. For example, there is bead region 1, bead region 2. On bead region 1, you have capture antibody for IL-2. On bead region 2, you may have capture antibody for TNF alpha. Those sort of things. And that is where once this are uniquely, the assays are defined, what bead regions are captured with what antibodies, and you put it on a software according to that. I'll show the in terms of the hardware and software how it is classified, the bead regions and the data coming out of for each bead region. Now you have two different dyes basically immersed. You insert the bead, it basically has its own unique spectral identification. And this is how basically it looks. On the left side you see 100 types of beads having different colors, just for graphical representation. And on the right you see the software. These are on the right, basically on the software you see the bead map according to this. For example, if you have 10 different targets, the 10 different targets or the targets or cytokines or analytes will be present in the 10 different dots mentioned on the software. And that is where you get your mean fluorescence intensity, that is where the data comes out. So in this technology there are basically 5 components to be precise. What are these 5 components? One definitely is your biological sample which you are looking at. Second is the beads, the unique beads which are provided from Bio-Rad. The third, fourth, fifth are the fluidics, optics and high speed digital processing. Dr. Vinay, the previous speaker, mentioned about hydrodynamic focusing, right, in terms of the flow. It is based on the same technology in terms of hydrodynamic focusing. You have the sheet fluid, you have the samples aspirated with the needle, and all the beads, whether it's a 10 target or 20 target, aspirated through that needle and going through this flow cell. And in terms of the architecture, there are 2 lasers. One laser basically identifies identifies which bead region it is. For example, bead region 2, 3 or 4. And the second laser identifies the mean fluorescence intensity in terms of the amount of target which is there. So that is how in principle this setup works for the bioplex 200. The workflow of this is basically on the left top you can see just the same as ELISA 96 well plate coming in. You have a concoction of beads depending upon the number of targets you are having, coupled with your capture antibody. Then you have in the second step you basically incubate and wash your samples with it. Then after that you make your sandwich immunoassay. You have your biotinetic detection antibodies captured. Okay. And once you do that, you add streptavidin PB for your excitation and capture of the fluorescence. The assay time basically it's around 4 hours. 45 minutes is the lead time for your requirement on the instrument. We do have as a part of workshop, we're doing one ticket for cytokines detecting this. Sorry. So this platform can analyze up to 100 different targets. Off the shelf, there are basically 48 plex available. It is not restricted to just this. There are multiple companies providing targets on this platform. It's an automated 96 cell plate. Each plate can run up to 80 samples in singlet. If you are looking at doing samples in duplicates, you may end up around 38 samples in each plate. So that is where in terms of you have to plan whether you want to do a singlet, duplicate, and number of plates required for each. Looking cost aspect also comes when we compare this to an ELISA, right? Because that is the key aspect. One aspect is if you are looking at targets beyond 4 cytokines or 4 analytes to be analyzed, this becomes an economical proposition, leaving alone the capital for procuring the instrument for a day-to-day usage of the things. It goes through a rigorous calibration validation which Bio-Rad has developed. Again, the signals are at least 5 order of magnitude when we compare to others, at least one log more in terms of getting the more sensitive signal from the data. Now the second platform again which Bio-Rad provides with the collaboration with Luminix is the 3D. It can have the option of 96 cell plate as well as 3T4 well plate. The good part is it can monitor up to 500 different cytokines in a single one. Usually these are being used in more of a diagnostic HLA typing applications per se. In research applications, whether it be biopharma or academic setup, we mainly see the Bioplex model being used up to a target of 20-25/7, which most of the academic labs use. But we do have the both platforms. Again, 21 CFR more catering to the biopharma space, whether they want the instrument to be in a regulated environment for having all the audit rate capture, whatever data has been done on the system. As I mentioned, this instrument throws out a lot of data, and data without proper interpretation is basically a garbage. And that is where Bio-Rad has developed Bioplex Manager, which not only analyzes but gives you a proper interpretation of this data. Because we believe that without interpretation, the amount of capital we put in and amount of effort we put in that doesn't make sense. And that is where Dr. Prashant is there. We'll be actually showing few of the examples of the data coming in, how do you interpret the mean MFI and things. You can just see the columns. If you run just 3 plates and 20 cell plates, you have 3078 points of data. And if you are doing at 50 plates, if you have running collaboration with a hospital setup project, 50 plates maybe in terms of catering to 400 samples, you have 51,000 data points. And getting those interpreted is a Herculean task. And the software helps you provide this platform. So we have 2 software packages or software catalog numbers to be precise. The Bioplex Manager collects, analyzes the data. It has something called a Brendan scientific 5PL algorithm for better curve fitting, which is there, which Dr. Prashant will just highlight on. And we have a Bioplex Data Pro, especially for the biopharma setup, for multiple getting data interpreted from multiple instruments in a single platform. So, be it whatever plot, be it a scatter plot, whatever format, the data can be thrown out in the different formats as and when needed. So these are the few of the examples which the data can be thrown out. You can see the normal breast cancer on the top, colon cancer, lung cancer, how these are represented in terms of standard as well as observed concentrations. Now few of the examples which Bio-Rad and this can be used across board. Now the theme of the workshop is tuberculosis. We have partnered with ICMR, that is Indian Council of Medical Research. You are aware that Government of India is on a war footing to eradicate TB from India by 2025. And we are glad that we have partnered with ICMR. There are studies going on this TB at 3 sites basically, in Chennai, that is NIIT, Pune, that is Nari, and AIIMS. So we have given the kits required, basically a 10-plex kit which is required, and they are doing studies for TB on this platform. And soon they in terms of, so that is where when Sir invited us, we were more than glad to partner, because we have partnered with ICMA for the TB eradication project which the Government of India has undertaken. Apart from that, as I mentioned, to monitor immune responses with Bioplex assays, especially not only from the academic but also from the biopharma. During COVID times, we partnered with Serum and other vaccine manufacturers globally. When getting out the vaccines early, you need to check the immunogenicity of the vaccines being rolled out. And we have partnered with them. They have got few instruments, at least many, and got out decent amount of data to access the immunogenicity measuring the antibody responses from the animal and mice model. to roll out to the next stage of the clinical trials. Also, streamlining clone selection in terms of quantifying expressions with multi-subunit viral proteins, which can be detected with the help of this. Aiding adjuvant selection also can be an option catering this Bio-Rad platform. Recently, we launched something called SARS-CoV-2 panel, the 4-plex. Also, we have launched something called an antibody for different strains, be it an alpha, beta, even omicron. So we have launched kits for detecting the antibodies developed if we have been infected with the different strains or different types of the coronavirus. So be it research and development, be it post-market surveillance, especially for the vaccine companies, you want to see whether after your vaccination or after this, what are the population having antibodies generated and till how long these are antibodies there, so that the booster doses or the next doses can be administered accordingly. So different the platform can be explored using this. To say the least, what are the applications which this platform can cater? We have partnered Luminex for especially the protein expression profiling. On the left, all the applications are mentioned over here. But this platform does not restrict you to do that. Luminex itself have developed application solutions not only for protein but genome research as well, HLA typing. Bio-Rad does not have solutions for these applications because we have not partnered it, but the platform which we are talking about gives you that flexibility. So it's not only the protein but also genotyping or genetic research which we can do on this platform. So that is from my end. I invite Dr. Prashant to hand over the session and talk about what are the softwares and the data which comes out and how we can interpret those data. Over to you.
Thank you, sir, for your valuable insights. Is there any questions?
Yeah. So one basically detects your sample that is immobilized on the bead, and the one is basically with a biotech antibody which becomes a capture, on which you have your detection or streptococcus cyclometry which is immobilized on it in the covalent required. So usually we say that anything above picogram levels could be detected on this instrument. Specificity basis, very specific, because we are having those capture antibodies specific to the antigen. So it's depending unless and until your samples are very crude and not treated properly. If you are looking plasma or basically those samples, we have got very good sensitivity across board for this across board.
Thanks.
Yes. So for example, if you are looking about 48plex, which is the example we showed, so there are 48 different kinds of beads immobilized with 48 different capture antibodies. All the ways. So we pipette it out. Basically, it comes in a vial, and then we pipette it out on each one. That has to do manually. It's a magnetic bead. It's basically a 6.5 micron magnetic beads immobilized with the targets which are looking at with their capture.
Sir, thank you, sir, for the talk.
Yeah.
Sir, I just wanted to know the limitation to this Bio-Plex. Does it have any limitations?
I would not say limitation. I would say first is yes, the investing in the instrument becomes a major challenge, because if we compare with LSR costing around in terms of 2 to 3 lakhs, with the Bio-Plex at least cost on a rough estimate 50 to 60 lakhs. That becomes the prime limitation or restriction, if I put it so, because putting this amount of money is a Herculean task, especially in the Indian setting for the last 2 years when the funding becomes restricted. The second part is If you're looking at in practical, the system can do up to 100 targets. But we have kits up to 48 kits. If you want to do beyond 48, for example, we just need to check the cross-reactivity of the specific antibodies which are there. That's the only, not restrictions, but the caution which has to apply. We have, bulletins mentioning what are the cross-reactivities which could happen, so that you select those antibody targets accordingly.
Thank you, sir.
Yeah.
Thank you, sir. Now I would like to invite our next speaker, Dr. Prashant Kumar Sharma, Application Supervisor, Bio-Rad Laboratories, with a great round of applause. Dr. Prashant earned his PhD from National Sharma Institute, Agra, is currently on Application Supervisor for Bio-Rad Laboratories, India. He specializes in Western blotting and Bioplex multiplex ELISA, instrumentation and associated experiments. He has been at Bio-Rad for 10 years and has been in the field of genomics and proteomics for the past 15 years. Now, over to you, sir.
So, good morning everyone, and I would like to thank Dr. Wayne to just give this opportunity to be here and present actually. So, already Amrut has explained a lot about Bioplex and has covered most of the part. So, I will focus on software and what are the precautions you should consider when you are doing experiments and when you are getting your data. Okay, how to interpret it. So data also I am going to show here, and again when you do the experiment, at the time we do it in laboratory form. Okay. So basically, whenever you are using any instrument, any sensitive instrument actually, so when instrument is good, okay, it will give you good data. Okay. But if you don't have any good software or the software which has multiple analysis feature, it is totally waste actually, because you can't interpret the data. So in that case, you need a powerful software as well. Okay, there are multiple techniques which can provide you good data, but if you don't have good software, you will not get that. So Bio-Rad has designed especially two softwares. Okay, so with Luminex, as Kumar told, this platform was designed by Luminex and being marketed by different companies like Bio-Rad and Multiplexing and other companies. Okay, but just to utilize this instrument in a better way, actually Bio-Rad has designed Bioplex Manager and Bioplex Data Pro. Two softwares are there. Okay, so these softwares will help you actually to interpret data in a better way and gives you freedom actually to analyze it for your further purpose. Okay, so this instrument is very good actually, or the data is whatever you are getting. Okay, so there are two things. So whatever disadvantages or whatever limitations you ask actually, so I will consider it as a limitation actually. So when you are getting data, okay, if you get very good data, it's good. But suppose if you are doing any mistake, that all will be cracked. So the system is so fast actually. If you compare it with ELISA or with any other technique, all other techniques are time taking, but it is so fast that within 4 hours you get huge data. Okay, so if you are doing some mistake in starting, it will totally crack. Okay, so but the software gives you freedom that you can utilize that data by omitting some outliers or some other problems which you face during run. So all those can be considered data also in the software. So these are the two softwares, Bioplex Manager and Bioplex Data Pro. Bioplex Manager is a single software which can control instrument and you can acquire data and you can analyze also. So with a single software you can do everything. Okay, and this was designed by Bio-Rad only. So this comes with Bio-Rad instrument also. And I have seen many customers who are using Bioplex instrument or Luminex system from other vendors, but they buy only this software just to analyze their data. So the software has so many features which other vendors are not providing. Okay. And the other software is Bioplex Data Pro. So Data Pro actually is for analysis purpose only. So suppose you are using Bio-Rad instrument, Luminex instrument or Multiplexing instrument. In that case, suppose you want to analyze data from their platform also with Bio-Rad, or you want to compare. or you are suppose from Bio-Rad itself you are using two different systems. One is Bio-Pix 200, the other one is MacPix. Okay, some customer have MacPix also. So both data sets will be different, but to analyze both data sets in a single software can be possible with the Bio-Pix data pro only. So suppose you are doing multiple experiments and you want to analyze all data together, you need to analyze it in Bio-Pix data pro. Okay. So it accepts data files from different platforms and different vendors also. Okay. So this is Bio-Pix Manager. So like as I told you, this is used for acquiring data, then controlling the instrument, for routine maintenance and for analysis purpose. So here actually and for troubleshooting also. So whatever experiments you are doing, in every experiment you are running some dilutions actually to make a standard curve. Because for any quantification you need a standard curve. So here for making a standard curve you are using like recombinant proteins which are already provided in the kits, and you are making dilutions, and then with those dilutions you are running a standard curve. So in normal softwares usually you get only linear standard curves, but here in this actually we are using Brandon scientific steps actually to implement 4PL or 5PL standard curves. Okay, so those standard curves can also be used in this software which are not available in the others. Okay, for routine maintenance purpose, this software gives you a lot of freedom, like when you are starting your experiment. So for a startup everything is there, so you just click on a startup, the instrument will be ready within half an hour. Then you have to like design your experiment. Within experiment you want to wash your plate between, that option is also there. After completing your experiment, routine maintenance you want to do, just shutdown option is there. So when you click shutdown, it will do all the maintenance, then only it will shut down. Okay, so it takes care of all the things actually whatever required. Generally with expert technology, whatever software is being provided, that doesn't include all these things. So there everything you have to do manually one by one. Okay. Here actually adjust needle option is also there. So whenever you are doing experiment, if your needle height is not properly set, in that case whatever sample you are putting in the web, that will not be aspirated, or like suppose your needle height is very low, in that case needle can clog and the sample can go in waste also. Okay. So all those things are there. Now this software can generate you data in different formats, which Amar showed you, like different type of graphs, like heat maps or like bar diagrams. You can, or different scatter plots. All those things are there, which can be generated in these softwares. So now, Bio-Pix data flow is there for analysis purpose, for better analysis purpose, and Bio-Pix Manager is there to control, to acquire, and to analyze data. Okay. And as I told, this Bio-Pix data flow can accept data from different platforms as well. So now, when you are using Bio-Pix data flow, Bio-Pix Manager only. So what are the things actually there which this software can provide, and what are the things you should consider when you are doing any experiment. So suppose you can run your experiment, you can get your raw data. You can change DD gates in this software. So when you are running your experiment, suppose you have done DD gates. So the DD is doublet discriminator. Okay. So when this gate setting is not proper, in that case, suppose your beads are lying in some other region, but you have done some wrong DD gate setting. In that case, what will happen? Suppose you are not there in front of instrument and you have started your run and you went just for a tea break. When you are coming back, then you realize that there is no data actually. In everywhere it is showing low beat count or something. So this can happen due to wrong DD gate setting. So this software provides you freedom to set the DD gates after experiment also. So suppose during experiment you have done wrong DD gate setting, or by default whatever DD gate setting was there, you have just run with that. Later also you can refine that actually. Okay. And excessive bead aggregation. If excessive bead aggregation is there, like when you are using your samples which is not that much clean, suppose you are using serum or plasma, so without centrifugation you are just taking samples, so lot of precipitate is also coming. In that case what will happen? Lot of aggregation may happen. Or suppose sometimes I have seen customers use neat plasma or neat serum. So when you are using neat plasma, neat serum, in that case lot of aggregation happens. Or suppose whatever sample you are having that has like very high load of different cytokines and chemokines, in that case also you will see lot of aggregation. Or suppose whatever regions you are using, all regions are very cold actually. You started your experiment without reaching it at room temperature. So when everything we are using very cold, aggregation you will see. So to remove that aggregation also actually in this software multiple features are there, by that you can reduce aggregation percentage. A small particulate matter tablet. So when you are running experiment, definitely during experiment you will see lot of small particles, like if there is any crystallization in the buffer itself, suppose, or whatever sample you are putting, and that aggregation is also happening, or some precipitate is coming during experiment itself. So that it takes care of that all SPD actually. Then bead counts and air spikes. So air spikes, suppose when you are doing experiment, your needle is not adjusted properly. So in that case, like lot of air can enter in the system, and then air will be like represented as some beads during when you are acquiring data. So you can differentiate it where is the air and where are the beads actually. So that freedom is also there. Bead count in detail, it tells you the bead counts, that how many beads are being aspirated, how many beads are there for each analyte actually. So suppose, as I will give example for 48. So you are running 48 different analytes in a single bag. Okay, so suppose there are 5 lakh beads. For each analyte there are like 1 or 10,000 beads are there. And from that it is aspirating and then plotting it on a scatter plot. Okay, so suppose your needle is not adjusted, in that case bead aspiration will be less. Second is how many beads it is aspirating, how many it is reading, how much percentage is aggregation, how many beads are there for each analyte, that also it is telling you. Okay, so suppose you are running 48, out of 48 you are getting proper beads for 40 analytes, but for 8 analytes you are not getting proper beads. In that case you can set time also. So when it is aspirating sample, it will keep on aspirating sample to read the beads from other analytes which are shortfall actually. Okay, so which are less in the count. So to increase that count, it will keep on aspirating and it will make the proper data in a single run itself. Otherwise, most of the time when you are setting a time for 45 seconds, within 45 seconds, if it is not able to aspirate sufficient sample, in that case what will happen? You have to read this sample again. Okay, so whatever you have read earlier, that is totally waste. So that also it takes care. So this is that DD gate setting. So this DD gate setting actually you can set. So suppose this black peak is coming, this is the region where your beads are coming. Okay, but if you are setting a DD gate in which this peak is not lying, then all data will be lost actually. So later if you set this DD gate, you can see that data. So that freedom is there. Now this software differentiate access the aggregation. So whatever small peaks you are seeing, those are aggregations. Okay. So this tells you how much percentage of aggregation is there. So in this, on the right side you can see 16% aggregation. So we usually consider a sample which is showing aggregation not more than 20%. Usually aggregation should be less than 20%, but if it is coming more than that, then also you can consider your experiment. Depends on different conditions actually. So whatever standard curve you are running, in that also aggregation is coming or not. If your sample is compatible with the beads or not. So lot of parameters are there, which we will explain later when we do the experiment. So why this aggregation comes, which I told you, highly viscous sample. Suppose you are using neat sample like serum, plasma or culture supernatant. Cold acid buffers, which I told you, insufficient plate mixing. When you are placing your plate, okay, so these are beads. Okay, beads are in a solution. If you are keeping the plate stagnant, in that case, there will not be proper interaction between antibodies and analyte and beads. So you need a proper aggregation, proper shaking actually. So here we do orbital shaking, and in most of the kit we do shaking at 850 rpm to 900 rpm. For some kits we do at higher speed also, but usually we do at 850. Okay, so that we need a proper shaker actually to shake the plate. Then excessive vacuum during filter plate wash. So means here Abhrat explained about magnetic beads. In this system you can use non-magnetic beads as well. So this system is compatible for non-magnetic beads also. So many customers who use filter plates. So there are plates in which at bottom there is a filter. So when you are doing your experiment, when you are using magnetic beads, in that case you use a magnet to trap all the beads and you can remove all the buffer from top. But suppose you are not using magnetic beads, in that case you need filter plates. So from filter plate, everything will pass through the bottom. So at bottom there is a filter through which everything is passing. So when you are using a filter plate for doing any experiment, in that case, suppose you are applying more vacuum or excessive vacuum, in that case it can rupture the seed, and you can see that lot of beads have been lost actually during washing itself. So that also you will see here that lot of bead lost and count is less. So earlier actually Bio-Rad was also there providing for these filter plate based kits, but now we don't recommend actually to customer because the recovery with magnetic beads is more in comparison to filter plates, because magnetic beads you can trap with magnet and you can do multiple washings easily. Now suppose when you are running experiment, you see all these red kind of dots when you are doing any experiment. So these dots looks like in this chromogram it looks like small peaks. So what are these things? Suppose for mixing your beads, you are doing sonication. So during sonication, if you are using high intensity or if you are doing sonication for more time, in that case, lot of breakage happens. Okay, beads are stored at room at 4 degrees Celsius. When you have to run it, you have to bring it at room temperature, then you have to dilute it, then only you have to use. Sometimes, when you are doing vortexing, okay, if it is recommended for 10 seconds, someone is doing vortexing for 30 seconds or 1 minute. In that case also breakage can happen, or sometimes in some cases it is recommended to sonicate beads for few seconds also. So if you are doing sonication with higher intensity or doing it for more time, you can see breakage of beads. So that looks like this in the experiment. Then air spikes on the right side, actually you can see this is the air spike. Suppose air is being aspirated through the needle, okay, because there is no other point from where air can enter in the system. Only through the needle air can enter. Now, so it is being just due to incorrectly adjusted needle. So in the software there is an option to set the needle according to different planes. So as when we are using magnetic beads, we usually use flat bottom plates, and for flat bottom plate we consider a standard plate only. When you are doing any HLA typing or any genotyping assay, in that case you use PCR plates. So for PCR plate, probe height setting will be different. So that also, when you are doing experiment, before starting itself, you should consider all these things, that everything should be set actually. Because after completing your experiment, after completing all your incubation, you have to place your plate in the instrument and then you have to read. Okay. So before reading, everything should be properly aligned. Because whatever is there for 50 microliter, from 50 microliter, it is aspirating 20-25 microliter. And if in first run or in first read you are losing a lot of things, then again will be difficult to recover in second run. So better to be sure about all the settings in the instrument. That's why I said, this is good system which provides a lot of data in just a small time. But it is bad also, if you do some mistake, you are losing a lot of things actually. So one kit costs around 1.5 lakh, 2 lakh, or more than that. There are some kits which are around 5 lakhs rupees. Okay, so within 5 lakhs you can get data for 80 samples or 38 samples. But suppose you are doing some mistake, all will be made. So here, when you are doing experiment, bead region also you should select properly. So in this software actually, our software provides you freedom to change the bead region later also. Suppose you are doing any experiment, by mistake you selected wrong bead region. So for each analyte there will be a different bead region. There are 48 analytes, so you have to use 48 type beads, and each bead will appear here in different segment actually, or different area. So if you are selecting wrong bead region, in this actually you can see on the right side, here this is wrong bead region, beads are not coming in this region, beads are going somewhere else actually, but we selected wrong bead region, that's why it is looking like that. For others it is correct, but for this it is not correct. Here there should be one bead region actually, but Here we have not selected any region actually. So we are losing beads. So this change we can do later also. Now bead scatter. Sometimes you will see a lot of bead scattering also. Okay, so this also happens when you are not taking care during experiment itself. So suppose you are doing a lot of shaking, lot of sonication, or a lot of breakage is happening. Okay, or suppose you are using some detergents or salt, which is changing the pattern of the beads actually. So when pattern of the bead is changing, the bead will appear at different location from its desired location. So that should not be done. So whenever you are optimizing your experiment, in that case, initially you can check all these things and then you can scale up your experiment for further samples. So like when ionic detergent you are using, or excessive salt you are using, or highly lipemic sample you are using, in that case you can see this bead is scattering. Okay. Suppose you are doing like amine coupling also when you are doing manually. So in that case, suppose bead like cross reactivity is there, in that case also you can see this bead is scattering. So there are multiple companies who are providing kits, okay, like Bio-Rad, but Bio-Rad provides you freedom to design your own kits as well. So you can buy your beads, you can like incubate it with antibodies, primary antibodies, it can be captured on the beads, then you can use that for your further experiments. So suppose like there are kits for 4-plex, 8-plex, or up to 48-plex. Suppose you want to design a kit for 4 or 5 targets only, and those targets are not available in any kit. So in that case, first you have to like buy 5 types of beads, then you have to capture all your monoclonal antibodies on those 5 types of beads. First you have to run independently, like in single well, just see the pattern, then you can mix them one by one and then see the pattern. So you are running single, then you are running in multiplex mode, and that way you can optimize that how your pattern is coming actually. If there is any cross reactivity, then you will not get any data, or the data will not come in that particular segment where it should come. So that freedom is also there, like you can design your own things. Now for doing like after doing any experiment, software automatically gives you a standard curve. So there are multiple options available for the standard curves like linear, 4PL, 5PL, and other options are also there. And it gives limit of detection, lower limit of detection. So whatever analytes you are running, actually that should come in between that. If anything coming beyond upper limit of detection, then it means you've done some mistake in your sample dilution, or there is something which is hampering the detection, proper detection. Just because of that it is coming more. So in this actually you can see that we are making 8 dilutions from S1 to S8. Okay, and this is 4PL, or like this is 5PL curve. Okay. So when your curve is properly sigmoidal, sigmoidal means it looks like S. So both upper and lower end are looking like mirror image of each other. In that case you can apply 5PL or 4PL curve. But when you are using any like sample or any standard where upper or lower points are not coming properly, they are being missed actually, in that case you can use linear curve as well. So it gives you freedom to select different type of like curves or different stats according to sample or according to analyte. Like you are running 4 analytes, for one it is giving poor results, in that case you can change the stats, and for other you can choose different parameters. So this software can give you data according to that. So here it is showing for one analyte, like it shows for each analyte. So whatever samples run for this, all are being shown here as a green color actually. So all are coming within the range of this standard curve. These yellow spots are actually controls. So controls are also coming within the range. Some are going out of the limit actually. So whenever you do your experiment, you get your data in this way actually. And when you are doing any optimization in the standard curve, it gives you the output that it is successful or it has failed. So when it has failed, then you remove any standard point actually. In that case, you apply the lower parameter, like in place of 5, you can apply 4PL. So for 5PL, you need at least 6 data points. So if 6 are not coming, in that case you have to apply 4PL. If 5 points are also not coming, then you will apply linear mode. So you can, even after doing a bad experiment, at least you can get some data. So you have done some mistakes during experiment, data was not proper, but even then you can get something. So after that actually it gives you the recovery here, so observed versus expected. So this percentage is actually the recovery. So this recovery should be between 70 to 120% actually. So whatever you are putting in the well, that is being recovered or not, actually that it shows here on the right side. And whatever CV is there between the replicates, that also it is showing. So CV should be as minimum as possible. And if, like suppose you are running duplicates or triplicates, out of them if any replicate is not working fine, you can remove that as well after like when you are doing analysis. So suppose you are running triplicate, one replicate is not working fine, you can remove that. So just because of one replicate, it can spoil your standard curve as well. So you can remove that and you can consider only two replicates. So that freedom is also there. Now after that, it gives you the recovery also. So the recovery percentage you can select according to your experiment. Suppose you are seeing that for every analyte result came properly. So in that case, you can select recovery between 90 to 110. But in most of the experiment, we consider between 70 to 130 just to cover everything actually. Okay, but you want to see only the precise data. In that case, you can decrease this range. So as a narrow range you are selecting, you are just focusing on the precise data. Okay, then next is concentration. So when like here we consider only FI value. FI value means fluorescence intensity. So for standard curve, whatever FI value is coming for any sample, if it is not coming in within that limit, okay, we don't consider actually that concentration. So even when for this sample some FI is being detected, like some fluorescence is being detected and it is giving you some concentration also. But this concentration it is showing with the asterisk, so it means it is being extrapolated and then only being calculated. So this is not coming within the range of the standard curve. So it gives you data, but it is up to you that you want to use it or not actually. So whenever anything is coming out of range below the lowest dilution or out of range above the highest dilution, it gives you some data but with some asterisk mark. And here it gives you the FI value also. So with FI you can cross check whether it is coming within that range or not. Now it gives you the concentration in range also and like observed concentration as well. So like it depends on the experiment or depends on your analysis which parameter you should consider. But most of the time we consider the FI value actually. So if FI is not coming within the range, we don't consider that. So suppose when you are seeing that when you are considering concentration in range or observed concentration, and if it is not coming within the range, it is showing OR lesser than the lowest dilution or OR more than the highest dilution. In that case, you can remove some standards and then you can see that your sample is coming within the range or not. Sometimes it happens actually. Now, to get the proper data, you need properly aligned instrument. So, like whatever software comes with Bio-Rad system by default, that is XMAP. So with XMAP actually whatever kit they provide, that is not doing that rigorous testing or rigorous validation. So Bio-Rad designed its own calibration and validation. So it covers a lot of parameters within the instrument, so that after doing this you are sure that instrument is working fine. Okay, so we do calibration. When calibration 1 and 2 passed, then it gives you reminder that whether you need to do validation or not. Okay. So if your validation has already failed or it has already passed the limit of 30 days, then you need to do validation as well. Or suppose you are shipping your instrument from one lab to another lab, you need to do calibration and validation every time. So this calibration is valid only for 4 hours when you are doing any experiment. So every day, whenever you are running experiment, you need to do calibration. Suppose your instrument is idle for 4 hours, you have not used it, you need to do calibration again. But validation you have to do once in 30 days only. Or whenever it shows that now validation is out, you need to do validation again. Okay. So in that case, this is the validation kit which we provide. So you can see it takes care of all the optics alignment, all the integrity of fluidics, like all the fluidics is working fine. So fluidics means whatever beads you are putting in the well, actually it is aspirating number of beads, like sufficient number of beads, and it is not cross contaminating the well. Suppose needle is going in one well, then aspirating, then next time needle is going to next well. So there should not be any cross contamination. So in first well there are beads which are having high intensity, in the other well we are having only beads, there is no analyte. So there should not be any data in the second well. So it takes care of all that actually. It checks that is there any cross contamination or not. So that parameter also we should consider. Now next is reporter channel performance. So as there are two lasers, green and red laser, so you have to check whether both lasers are working fine or not. So there are some beads actually for reporter validation, and then classification also. Classification means whatever beads you are putting, it is classifying properly. Like it is suppose you have selected 4 different regions, it should classify 4 different regions very accurately. And when it comes to validation kit, whatever regions we have selected in this validation kit, those are very close to each other. So like if it is able to select all the close regions very accurately, it means the instrument working fine. So every day like we do calibration, but validation once in a month or whenever it shows failed. Okay, so after validation you can see the report as well. So when any validation parameter is not coming within the range, it gives you result just by that phone that it is not passing. So you can like do some cleaning of the instrument, cleaning of the fluidics, or you can check whatever parameter is not coming within the range. So what could be the reason actually, you should focus on that. Suppose your optics is failing again and again, or your classification is failing again and again, then it means there is something in the optics itself. So you need to do laser alignment. So laser alignment should be done by engineer like before doing any further test. So to verify all those things, kits are there, you should use them, then only you should do your experiments. Now, thank you for your attention. So till now, you have any doubts actually, or you understood basic actually what is BioPax technology? Because after this we will do one experiment and you should be clear actually about concept. Oh, without that also you are audible. For it? Yeah. So in this kit actually they recommend that at how much amplitude you should do the sonication only. We need only mild sonication, like you can use ultrasonic cleaner. We don't use probe sonicators because those are more intense than ultrasonic cleaners only. So we have to just remove break the aggregates. So when you are storing beads at 4 degrees Celsius or at lower temperature, all are then actually aggregating, and when you keep it at room temperature also, those aggregates are not breaking. So when you run those beads, you will not get proper data. Suppose you are adding only 5,000 beads per well, and out of 5,000, 4,000 have been aggregated. So only you have 1,000 beads free. Okay, when it is aspirating, so in aspiration all the aggregates are being aspirated first, suppose. So when it is aspirating aggregate, it will clog the needle. So when your needle is clogged, then it will not aspire other sample or that remaining sample. So just to avoid that actually you need to do sonication or proper vortex. So that in the bead type wherever aggregation is more, they recommend sonication and they mention in the kit manual that for this particular bead type you need a sonication. So for every bead type it is not required.
Anyone else?
So about software actually, should we start experiment now, or like I can show the software as well here? Okay. So basically this is the software. So let me show it from starting. Bio-Rad Manager 6.1. So this is there are two types of softwares we provide. One is for instrument control and one is for analysis only. So this software, actually there is some dongle. One red, this dongle you can see. So this dongle is actually for using this software. Without this dongle, you can't use this software. So this dongle is for instrument control as well. That's why it shows attempting to run the instrument. Okay, but we can use it for analysis also. So here I am just opening one data set. So in this software, for opening any data file, we need only RBX data file or CSV data files. So suppose you are running your experiment with XMAP software. So with that software, you have to export your data into CSV format, and then that CSV file you can open in the software. Otherwise, if you are running through Bio-Rad Manager, you can directly open this. So after running, like you can see here, this is raw data. So here there are only like 8 analytes in this kit. So in this these 8 analytes, you can see all 8 are coming at proper location. First thing, there is no air, only small breakage is coming. Okay, so like very good run. And now we have to see whether bead count is coming proper or not in this run. So for each analyte, you can see for each sample that this bead number. So the whatever the number within the bracket, actually that is bead number. So minimum 50 beads should be there actually for considering this data for any analyte. When you are starting your experiment, initially itself actually you can define that you need 100 beads or 50 beads or 75 beads minimum. So for any analysis, we consider minimum 50. So at least we should consider that. So for each lane, actually you can see for all the analytes, it is minimum 50. Okay. So for some it is going more than 50, but for most of them, it should be minimum 50. Now it is showing that how many beads it has considered. 416 bead it has considered. So if you pool all the beads, actually 51, then 50, then 51. It will be 416. So total 416 beads it has read. Within the gate, 453 came actually. So like whatever beads used for analysis, there were only 453 beads. So there were some beads actually which which were not from these regions, or which were some like broken beads or aggregated beads. So you can see there 13% aggregation is also there. So there like some beads were rejected due to aggregation as well. So for each sample you can check these parameters whether your bead count is proper and aggregation is low or not. Okay. Now, so yeah, bead number is more because it is considering more beads. Suppose any aggregation is also coming within the gate, that will also be considered. Yeah, that will increase the number. So for every aggregate it will consider only one actually. In that case it will reduce the count actually. So suppose here more number of beads it is considering means lot of aggregates are also there. So that's why we lost lot of beads. But even then 416 beads it is like able to consider for analysis purpose. Okay. So these are standards actually. So you can see the dilution pattern which is coming. So this is Fi data only, fluorescence intensity data. And you can see this is gradually falling. So like we have done 4-fold dilutions, and with every 4-fold dilution the intensity is coming down for each analyte. Sorry. Because you are diluting. So initially the first one, S1 is the highest concentration, then you diluted it 4 times, then you again diluted 4 times. So like that you are diluting. So gradually it is coming down. So we have done this these standards in duplicates. So you can see one series from S1 to S8, then again S1 to S8, then blank. So every time you are running blank also in this experiment. So for blank actually If you are using serum samples, then you should use like serum diluent as a blank, or you can directly use serum, plain serum also, okay, or diluted serum. Or if you are using culture supernatant, then you should use culture supernatant as a dilute, because blank you are using for background correction. Okay, so some background will come in each well, so that background should also come in the standard well. So sometimes when we dilute standards with the sample as well. So suppose you are using culture supernatant, so whatever media you have taken in the cell culturing, that media should be used for making dilutions. Okay, so that background should be constant in samples and in standards. Now if you see lower actually, so there are different type of samples, so that marking you can do. So initially also that marking you can do in this description box, or later also you can do. Okay, after seeing all this, now you can go to report table. So this is the report table. So in this report table actually you can see that what is the bead count considered and what is the recovery actually. So this recovery, whatever you are considering, you can set in the next tab, standard curve tab. So here you can set the recovery. So 70 to 130 we have considered. Usually we consider within this range, but if you want to select any narrow range, you can select that also. So this recovery it is showing here. So within that range, like within 70 to 130 it is coming. Now when your standard data is coming okay, then you go to your sample. So you can see for most of the sample, for this analyte actually, it is showing out of range below the lowest dilution. So out of range below the lowest dilution, if you see standard curve for this particular sample, all the samples are coming at bottom. So all the samples are coming at bottom means the percentage or the availability of this particular analyte in this sample is very low. If you go to any other analyte, just check for other analyte. So for this also it is very low. Go for some other parameter. This is coming within some range. So this is also coming within the range. So this is coming between S7 and others actually. So other higher. So the concentration is quite enough. If you go for graph pattern, now you can see few samples are having high concentration of this analyte. And if you compare this with standards, so in comparison to standard, this is coming between S4 and S5. So for many samples it is coming between S4 and S5. So this data actually you can use for your further experimentation or further analysis. Yes. Yeah. So you can go to this report table, and from here you can select standards and you can remove them actually. So in standard curve also, like if you are selecting only 4 PL, then in that case you are removing some standards. Okay. So with that also the data will change like this. So you just see the samples only in that case. Okay. If your standards are very high, in that case you can just see the samples. You can remove the standards totally. So this way actually it gives you all the data for each standard. Now suppose for analyzing any analyte, suppose for this standard is very good, but the upper standards are not that much accurate as lower actually. So lower are coming proper S type, but upper are not coming proper S type. Okay, and upper is the highest, which is coming as upper limit of detection. So when it is reaching up to upper limit of detection, whatever sample comes above that actually it will not be considered. Okay, and it can give the data after extrapolation only. So for most of the sample it is coming within the range actually, within this standard curve range. Many times we use only this linear range for analysis. So if you are selecting only linear curve like this, okay, linear, so then in that case you can select only linear range. So if samples are coming within linear range, then good. If they are not coming within linear range, then you go for 4 PL or 5 PL like this. So and same regression type, like you are selecting 4 PL for this analyte, same regression type you can apply for all the analytes. So you can click here and you can apply this. Now this is already optimized, so I can see the report, or I can optimize it again. So when you are optimizing, it is telling you it is successful. Okay, so suppose if any standard which is not coming within the range of this type of 5PL standard curve, it gives here some comments that one standard removed. Okay, so suppose if you want to consider the data even without that, you can use that. So this way it gives report for all. Now if I undo optimization, again it will be raw data. If I optimize it, it is optimized data. So this optimization feature is also there. So it is not required that you do optimization for each and analyte one by one. You select one type of parameter, like you suppose you want to apply, first of all 5PL is the best, so everyone want to apply 5PL. So select 5PL, then same regression type for all analyte. Then here you can see, like you can tick all these, otherwise you can remove them, and then optimize for all. Then you can see the report actually. So optimization report is there. So this much analytes we have run. Now you can see some are optimized, some are not optimized. Okay, so now you can do optimization for all, undo optimization, then apply same, then apply across all analyte, then optimize. So now it is applying for each analyte the same parameters. So for two analytes, only one standard has been removed. For rest all the standards are working. Okay, so means this, all the experiment whatever you have done, it is good actually, because all the dilutions higher and lower all are being detected and all are coming within the 5PL range. Sometimes due to a lot of aggregation it happens that you lose lot of lower concentration or lower dilutions. So after doing this optimization, you have to see just your report table, then you can see concentration for your samples. So for any sample, if it is showing concentration with asterisk, it means it has extrapolated for that data. So here for this particular analyte, it has not like extrapolated anything, everything is coming within the range. You want to see within standard curve, you can just see here, all samples are coming within the range. Okay, so this way actually it provides you all the data. Now after doing experiment also, you can do all the labeling. Suppose here whatever labeling you have done that is wrong, so you relabel the samples, again the interpretation will change. Suppose you have done standards in last column in place of first column, that you can change. You have placed blank in place of in these third and fourth well, you placed on these wells, you can change. So everything can be changed actually. So it gives you that freedom actually to change all those things. So when you are doing ELISA also, you are getting this concentration only, observed concentration. Here also it is giving you the data, observed concentration directly. So after getting this data, you can export this to Excel, and if you want to use any other software actually for analysis, here you can export it to Excel and then you can use it. And it gives you data in multiple formats like single analyte format, multi-analyte, or 96-well format. Means in single analyte format, in each Excel sheet only one analyte data will be there. In multi-analyte, in single Excel sheet everything will be there. In 96-well plate, in plate format data will be there. So it gives in different formats actually, which are compatible to other softwares as well. So you can see here concentration in range and observed concentration both are same. Okay. So here actually this is like, just a minute, just let me see the plate pattern. So for each well actually, each sample is in single well only, it is not replicate. It is not replicate first thing, so each well is in single well. Now when you are seeing table, here there is option to remove the replicate, so I am removing that. Now you can see only for one. So if you are running in replicate format, you just click here. So at highest point it shows for replicate and then individual as well. So for standards we have done replicates. For sample it is not replicate. Yeah. So like in first one it is showing average and then individual as well. So suppose if any replicate is not coming proper, you can remove that actually by just selecting it. So I think it is already 12:30. Now you are here for next 2 days, so you can discuss any other queries also.
Thank you, sir. Hello. Hello. Thank you, sir. Now I would like to invite Dr. Ved Prakash Trivedi to facilitate Dr. Amrit Raj and Dr. Prashant Kumar Sharma. So now you all are invited for lunch and one more. And after the lunch, there will be the parallel sessions on flow cytometry and bioplex in the lab, along with the sample preparation, acquisition, and analysis. I request the participants to join us in the lab after the lunch. And one more thing, the candidates are requested to reach from tomorrow by 8:50 a.m. sharp, because there will be important lectures from tomorrow scheduled at 9 a.m. So you all are requested to please reach here by 8:50 a.m. Thank you.